sound.
बॉक्स ठेवलंय का? बरं बरं एकदम मस्त. 1-2 वाजेपर्यंत होऊन जाईल सगळं. लंच टाइमला सगळं मोकळं. उमेश सर आले तर भेटले का नाही? नाही भेटले का नाही? लाईट लावून टाक सगळे. नाही त्याची काही गरज नाही, ते नंतर करता येईल.
आवाज येतो का?
आवाजच येत नाही कोण तिथे येणार. ते साऊंड सिस्टीम त्यांनी काढली असेल. साऊंड सिस्टीमचा कर ना.
झालं का सुरू? हॅलो मॅडम, आवाज येत आहे?
येस सर.
हा मॅडम, तुमचं हे लाईव्ह रेकॉर्डिंग आहे त्याच्यामुळं सर नंतर बघतील म्हणजे तुमचं, तर तुम्ही चालू करू शकता आता.
ओके सर, कॅन आय शेअर माय स्क्रीन सर?
हो प्लीज.
Okay sir. Sir, but I unable. Okay, only the host can share this meeting, sir. I
wait.
unable to share. Yeah. Yes sir, now I can share my screen, sir.
त्याचा आवाज येत आहे?
Yes sir. It is there, sir?
Yes. You can
Okay,
proceed.
Okay, thank you sir. Thank you for giving this opportunity. I Vidya Maga, presenting my poster on molecular.
Your screen is not visible. It's just Vidya Magar, nothing else.
Okay sir. Again, I will try, sir. Just wait. So, okay. Sir, again, I will try to share my screen. Sir, it is there now?
No.
Screen share hot nahi hai. Okay sir, just wait sir for 2 minutes just sir. Share hot nahi hai screen.
You can send your PowerPoint so that I can share it to the group, and you can just talk on the PowerPoint like this. We can do.
Okay sir, I think it is there now.
Yes.
Okay, so can I start, sir?
Yeah, sure.
Okay, thank you sir. So I Vidya Magar presenting my poster on molecular docking studies of novel pyrimidine derivative on enoyl acyl CoA protein under the guidance of Dr. Lalit Sonone sir. This is my content of presentation. As we know, tuberculosis is a leading cause of death, and this is because of resistance of mutated Mycobacterium tuberculosis. So we can use advanced technique like computer-aided drug design to find out lead which will be more effective against this mutated Mycobacterium tuberculosis. Now, mycolic acid is important component of bacterial cell wall, and enoyl acyl carrier protein is important enzyme involved in this. So this enzyme, you can see here, it catalyzes this important reaction where it reduce double bond and convert enoyl CoA into acyl CoA. So this step is important in mycolic acid synthesis. So we can target this enzyme and we can inhibit mycolic acid synthesis. From literature review, it is found that tyrosine 158 is important amino acid located on active pocket of this enoyl CoA. And it has two conformation. That is why in our study, we have done 3 PDBs. We have taken 3 PDBs and we have done docking. So in material and method, we have taken this conjugate. So benes, isoxazole and pyrimidine, these two nucleus, both found as antimicrobial. So to get more effective antimicrobial agent, we have combined this in one nucleus. And this is our pyrimidine derivative. So this R1 is heterocycle. So here we have substituted various heterocycle like furan, thiopine, pyrimidine, pyridine. And this R is, actually it is X, hetero atoms are there. So we have substituted nitrogen, oxygen and sulfur. And with this, we have prepared 330 compounds, which is pyrimidine derivative. So structure-based drug design we have done. If we know structure of protein or target, and if we know amino acids which is located on active pocket, then easily we can dock and we can find interaction of our interested derivatives. So in this, we have used 3 different PDB ID. In that, first is 5-MTP. This co-crystallized structure we have used for docking purpose. So you can see this I have taken from PyMol software, which is free software. So I have sketched in PyMol. You can see here interactions also. That dotted line indicate hydrogen bonding of this amino acid 158 and glutamine with this inhibitor. It is co-crystallized ligand is there. Inbound ligand is also there. That is why I have selected this. Similarly, I have selected 4-COD, which having inbound ligand inhibitor. And 6R9W also I have selected. So this drug likeliness property also we have selected. So as we know, there should be not more than 10 hydrogen bond acceptor, not more than 5 donor, partition coefficient not more than 5, and solubility also we have done, and molecular weight, it should not be more than 500 Dalton. So this drug likeliness property, you can see, it obey this all rules, and maybe this compounds orally active. So molecular docking we have done. So molecular docking is basically performed to find out interactions of ligand with receptor. So molecular docking answer mainly 2 questions. First, whether ligand interact favorably or not. And if it interact favorably, then what is preferred orientation? So we have done this molecular docking with Schrodinger software, that is Mestro. And basically there are 3 steps. That is preparation of ligand, preparation of protein, and then docking we have done. So this preparation of ligand is very simple. So in Mestro itself, we can draw the structure in 3D sketch, as well as we can draw in chem sketch, and we can save in mole format. And after that, we have to go for energy minimization. So that is called preparation of ligand. So I think there is some background sound. So please mute other participant. So preparation of ligand, in that 30 structure we have drawn, that is base imidazole nucleus. And in preparation of protein, in Schrodinger itself, there is protein preparation wizard. So as we know, this is X-ray crystallographic structure we have taken from protein data bank. And as there may be missing residue, missing loop, missing hydrogens. So with the help of this protein preparation, we can add polar hydrogen, we can add missing residue, missing loop, primer also we can add. So with this, we have prepared protein. So 3 PDB ID we have taken and we have prepared this. And once your protein is ready, once your ligand is ready, then easily we can upload this to file in doc model and we can just run this software. Depending upon interaction of ligand with receptor, we'll get docking score. So if interactions are good, then we'll get more negative score. That is indicate stable interaction of drug, or it is now not drug, it is ligand with receptors. So depending upon that, software automatically run and gives docking score. So as I have discussed, this tyrosine 158 has 2 confirmation, in and out confirmation. That is why we have overlapped this all 4 PDB and we have seen confirmation. So here you can see there is little bit change in confirmation of tyrosine 158. So this is docking score of pyrimidine derivative, that is that 30 structure we have dock, and you can see 5-MTP, 6R, 6W and 4COD. On that we have done study, and this colored box indicate topmost compound having good docking score as compared to others. And inbound ligand also you can see we have done validation, you can see optimization. That is mean by inbound ligand we have removed from that active pocket and again we have redock it. That is called validation. And that dock pose we have overlapped with inbound ligand pose, and it is found RMSD less than 1. You can see 0.6 is there, 0.4 and 0.7 means that is called validation. And 13, 14, 15 you can see having good docking score. Similarly 13, 14 in second also there is similarity, and in third also 13, 14, 15, 17 and 19 found good docking score. So this based compound we are going for synthesis and then activity, and then we are correlating docking score with actual biological activity. So this work is in proceeding, it is not done yet. Only docking we have done and synthesis now in progress. And this is docking confirmation. So for time limitation, I have only pasted one confirmation here, and this is benzimidazole moiety. So you can see here there is interaction with this amino acid. So there is glutamic acid 210, which is involved in interaction phenylalanine and tyrosine 158 also there. Okay, so here there is interaction. This is confirmation. And in conclusion, we can see this computer-aided drug design, which can reduce our time also, time for drug discovery, which is complex process, and which can identify inhibitors, which will be more effective and act as an anti-tubercular agent. So these are references, and this is my poster. So thank you very much, sir, for giving me this opportunity. Sir, are you there?
Thank you, madam, for wonderful presentation. तो गंथा है रे?
Sir, if any question you can ask.
Madam,
Anyone has?
Yeah, I
Yes,
think this is everything on in silico experiments, right?
yes, yes, sir.
Did you go for the in vivo or in vitro like that? Is it still
Uh, s-
important in vitro experiments? I think you have synthesized some pyrimidine derivatives and then you go for some enzyme inhibition experiment like that.
Sir, in M-Farm, actually, I have done this docking, synthesis, and activity. So there, I found docking score and actual bioassay is matching. But now I work is 30 derivative I have prepared, sir. It requires some time. Actually, 10 I have prepared,
Okay, okay,
and synthetic work is in progress, sir. After that, I will go for activity.
okay. Good
Yes.
luck, madam. Okay, so
Thank,
thank you, madam.
thank you very much, sir.
Okay, next.
pre-digested foodstuffs packaged in their cells. They are diverse processes, economic gain technology, which includes
Good morning, one and all. Good morning, one and all. We are in a third day of International Conference on Advances in Bioactive Molecules 2022. Hope you enjoyed the yesterday's sessions, which are fruitful and joyful for every one of us. Now, I would like to request to welcome Dr. Hanman Barkate sir, Senior Vice President and Global Head Medical Affairs, Glenmark, Mumbai. Sir, please come on the stage. Huge round of applause. I would like to request Professor T.A. Kadam sir to please introduce our today's first speaker, Dr. Hanman Barkate sir. Sir, please.
Good morning, everyone. I welcome Dr. Hanman Barkate sir. This is third day of international conference. Most of the speakers, day before yesterday also, and today, purely from academic backgrounds. Today's speaker, Dr. Hanman Barkate sir, he is by profession EMD pharmacology. And presently he is working as a vice president and global head medical affairs at Glenmark Pharmaceutical, Mumbai. He has more than 23 years experience of post-MD experience of working in large biopharmaceutical organizations in leadership role in function of medical affairs, clinical development, pharmacovigilance, medical information, and drug regulatory affairs. 2 years of experience of working as assistant lecturer in internal medicine, that is pre-MD, and assistant professor pharmacology, post-MD in Government Medical College and Hospital. Recipient of Chairman's Excellence Award in 2019 in Glenmark for building capabilities of medical affairs and creating values within and outside the organizations. Co-created and co-implemented clinical development and regulatory approval strategies for first to launch of PVR in India for COVID-19. Strategized and implemented scientific launch excellence of PVR in India and MEA. Extensive experience of presenting new proposals for marketing authorizations, clinical trials applications to scientific communities of various regulatory authorities, expertise in clinical development, regulatory approvals, commercialization of the biosimilars, NCS, and NDDS products. Then extensive experience of successfully launching new molecules, a few first time in India, MEA, Asia, Pacific, and Europe. Experience span multiple therapeutic areas like cardiovascular, diabetology, neurology, psychiatric, gastroenterology, urology, vaccines, oncology, dermatology, pulmonary, critical care, rheumatology, ophthalmology, and gynecology. More than 50 scientific publications on his record in index journals of repute. He has given many oral presentations at national and international conferences, review of Journal of Pharmaceutical Medicines, Springer Group. He remained invited speakers in national, international conference, World Biosimilar Congress in Switzerland in 2018, 19, 20, 21. So such personality is with us and he will deliver his talk. So I hope most of you will actively participate after his presentation also. Thank you. Barkat sir, please.
Good morning to all of you. So I want me to remember in the introduction as a physician, clinical pharmacologist, and a researcher. If you don't remember anything beyond this, it's absolutely fine, right? And today I'll be speaking about a topic which is very relevant from a research point of view. And I'm very happy to be part of this, the international conference on advances in bioactive molecules. So I was just wondering under this conference, what I can speak about, right? And then I could relate that the area wherein I'm working, particularly area of biologics and biosimilars, it is a part of bioactive molecules, in fact, right? So you start with the bioactives. From bioactives, you take selected bioactives and develop as a biological drug. And because of the cost constraint, so you try to develop a biosimilars, which will be more cost competitive to the biologicals. So that is the reason I selected the topics. I keep on traveling across the globe, do participate in a lot of international conferences, particularly in the area of clinical development, then pharmacovigilance, and medical sciences. So I'll be sharing my insight, and those insights are particularly gained through the experience which spans across 23 years. So don't go by my face value. That will not be commensurate with the year of experience, right? So the views which I'm going to share with you, these are all my personal views, and it has nothing to do with the organization wherein I work. And the experiences are based on my hands-on experience. And just for your information, I have been working in a biotech and pharma company, particularly on biologicals and biosimilars. As I look at your faces, I think I need to explain what does it mean by biologics and what does it mean by biosimilars. And I'm hopeful that you are already aware about the bioactive molecules as you have been attending this conference since last 3 days, right? So I have worked on a 13 biosimilars. Some of the biosimilars were world's first biosimilars. And my role, you will be wondering, I said I'm a physician, clinical pharmacologist, and a clinical researcher. So obviously you might have a question that what a medical doctor will be doing in a biopharma and the pharmaceutical industry. So my basic role in a pharma industry, biopharma industry is to take the molecules from a lab to a bench or lab to, you can see, the clinic. That is my role. So you develop a molecule, say for example, you have a very active bioactive molecules you have obtained from microbes, and you have some hypothesis that it could work as anti-diabetic. Then how to verify that? So I have that role to verify and get it validated. So how do I verify and validate it? I do a lot of the sequential research. And in that sequential research, and hopefully because of this COVID pandemic, all even the ordinary citizens are aware about what is a phase 1, phase 2, and phase 3 studies. And all this thanks to the media, which try to educate a lot of people on the vaccine development. Like particular vaccine is just completed phase 2, enter into the phase 3. So I'm hoping that you are aware about that, but still I will clarify what exactly done during those phases, right? So as I was talking, so my role is to take that molecule from lab to the clinic. And it takes a long time, almost 8 to 10 years to take that molecule. Only in case of the international emergency like COVID, that path get accelerated or expedited. Otherwise, it takes to 8 to 10 years of time to develop a molecule from lab to clinic. So as far as the scope of my presentation, I generally don't call presentation. I believe always in a dialogue. So anytime you feel that you need to ask question, please stop me and ask question. So I'm not a keynote speaker, right? If you understand, keynote speaker means you are not expected to interrupt him. You are not expected to ask the question, right? So it is a contrary to that. You can ask question anytime. If you are not clear about any aspect, you do reach out to me. So I'll be talking particularly in terms of, I'm not spending much time on the bioactives. As I said, you are always already well versed with the bioactives, but definitely I will speak about the biologicals and biosimilars. What is the difference between a biological biosimilar, and what is the difference of a bioactive from a biosimilars and a biologics? And then I will focus on particularly biosimilars. And this is a very interesting one. And then I will talk about the historical development of biologicals or biosimilars. There will be a lot of the medical terms during my presentation. So just pardon me for using those terms. And then how this clinical development of biosimilars is taken. As I said, if there is a molecule synthesized in lab, how do you take it to the clinic? So I'll be talking about that. And then how we get the marketing authorization or approval to commercialize that product. And lastly, I will conclude. This is my presentation. Can you make a slideshow, or it is already slideshow? Okay. So yeah. Okay. So bioactive molecules, you're aware that these are derived from a natural sources. That natural source could be plant, could be animals, or even it could be a microorganism. And I happened to listen to one of your the session during this conference. Dr. Yogesh was talking, and he was talking about the bioactive molecules obtained from a microbes, and even from the fecus, you can have the bioactive molecules synthesized. So this is quite interesting area, and there are a lot of advances happening. So these are the molecules which are obtained from plants, animal, and microorganisms. And the Indians, they have all the knowledge about the bioactive molecules. Only thing, it has not translated into the products which can be used by the general population, right? So these molecules can be like growth factors, angiogenic factors, cytokines, hormones. They're all part of bioactive molecules. Like we have a growth hormone. Growth hormone is in fact a bioactive molecule. It can be obtained from humans. It can be obtained from animals, and they are used for a therapeutic purpose. When they are used for a therapeutic purpose for treatment of any disease or prevention of disease, that becomes a therapeutic intervention. And of late, the plant-based bioactive molecules, they are getting a lot of importance, particularly in a treatment of a cancer. And they are having a lot of study done, and they have shown that the bioactive molecules, when they are used along with the chemotherapeutic agents, and you are aware that any cancer patient, when that patient is put on chemotherapeutic agent, the first serious adverse event, which is the experience, is a loss of hair. In a medical terms, we call it alopecia. And that is a very common side effect of chemotherapeutic agents. So these bioactive molecules have shown to reduce those kind of side effects. And that is the reason there is a growing interest in developing a plant-based bioactive molecules not only for cancer, even for diabetes. And again, in your conference, I have seen a lot of topic focusing on the molecules. I happened to listen to one of the faculty from CDR also. He was talking about a lot of bioactive molecules identified for a treatment of a diabetes. So these are quite interesting. A lot of development is happening in terms of bioactive molecules. Now moving from a bioactives to the biologicals and biosimilars. So what is done in by biologics? Biologics means these are the molecules whose active ingredients are derived from the animals, plants. It is bioactive molecule only. Then what is the difference? Difference is they are primarily manufactured with a recombinant DNA technology. That's the only difference. You obtain the bioactive molecules, and then by using a recombinant DNA technology, you try to develop in a large scale a product that is called as a biologics. So say I will give example like insulin. The insulin gene from humans is obtained and it is inserted into a plasmid. And from plasmid, it is inserted into a bacterial cell. And this bacterial cell will able to produce human insulin. So that is what is a recombinant technology. So in case of biologicals, it is the gene modified technology, which is helping to have a large scale production. Otherwise, you can't have a large scale production. You can use that product only in a lab testing. But if you want to make a use of that product for a general population, then you have to develop in a large scale. And that is the science of recombinant technology, right? Insulin. So earlier the insulin was obtained from animal source, porcine insulin. Now we have the human insulin. How human insulins are synthesized? They are synthesized by using this recombinant DNA technology. So for your understanding, biologicals means they are basically bioactive molecules, but they are subjected to recombinant DNA technology. And I have explained the recombinant DNA technology because there is no rocket science in it. Understanding recombinant DNA technology. Even 8th grade, the student have the definition of recombinant technology. So that is the biologics. And as far as the biologicals concerned, they are very costly. And if you understand, I can give examples like insulin. Insulin is a biologics. Then there are monoclonal antibodies, MAPs. A lot of MAPs are used for a cancer treatment, like rituximab, biacizumab, adalimumab. These are all products which are intended for a treatment of various diseases. They are biologics. And the biologics are different from a chemicals. Chemicals in the sense paracetamol. You are aware about the Dolo, and the COVID has made the Dolo very famous. It's a household name now. So that is a paracetamol. So paracetamol is a chemical, which is very small in structure, whereas biologicals are, these are the large molecules, very complex to manufacture, very unstable. And if you understand, the now shift is happening from chemicals to biologicals. All the difficult to treat diseases, the chronic diseases, the chemical compounds are not found to be that effective. You require a biological products. And globally, the data of biological markets in a 2021, it was a 302 billion USD. 1 billion equal to 3,000 million, and 1 million is 7.5 crore. So that biological market is approximately 2.3 lakh across the globe. And it is expected to get double in the next 5 years. So that is the potential of biologicals. And a lot of companies are focusing on the biologicals. And biologicals are all developed by biotech companies. And during this pandemic, which companies got highlighted? Was it the pharma company or biotech company? Who developed the vaccines? In India, you are aware, it's a Serum Institute. It's not pharmaceutical company. It is a basically vaccine manufacturer. Vaccine is again a biologicals, right? So they have taken a lead to develop all the vaccines for a treatment. And that's the reason internationally, as well as in India, all those biotech companies played their crucial role. At a global level, it is a BioNTech or Moderna. They provided the vaccine at a global level. It was not a Pfizer. Pfizer is world's #1 pharmaceutical company. It was not a Roche or AstraZeneca, but it was a small biotech companies. And here the scale is not important. It is a technology important. So they were able to developed this technology, and that technology for vaccine manufacturer was passed on to bigger companies like the Serum Institute. Did Serum Institute develop this vaccine from the lab to, you can see, the commercialization? Yes. Who was the developer of the vaccine, the Covishield in India? Serum was manufacturer, but who actually developed that? Right? It was a university in UK. It was Oxford University. They developed that, and they licensed it to manufacture to Serum Institute in India. They could manufacture, but the technology research work was done by the institute. Same was the case for the vaccine which are developed for COVID at international level, like BioNTech I mentioned, Moderna. These are small biotech companies. So they're developed and outsourced to the bigger pharma companies, and then they could manufacture it. So that is the role of technology, know-how, understanding. Now just moving to the biosimilar. So what is a biosimilar? Biosimilar is basically a biological product that is highly similar to approved biological product. So it is a similar to biologics, but it is not exactly same as that of biologics. It's like a generic. You know that there is an innovator product, and then a lot of other company try to copy that. That becomes a generic. So biosimilar means it is a generic of biologicals, which is a similar but not same. There is a difference between similar and same. It is a similar to the originator. So biological is the originator product, whereas the biosimilar is a similar product that of the innovator. There are a lot of the biosimilar products which are going off patent in the sense. So I can give you example, like for a treatment of rheumatoid arthritis and cancer, there is a brand name called as Humira. Humira is the world's top selling biologicals. And what would be its a sale? It's a 20 billion USD. So 20 billion USD, if you approximately translate into Indian rupees, it becomes a 1500 crores. That's a single brand. And these biologics are going off patent. When these biologicals get off patent, that is opportunity for other companies to develop a biosimilars. And that is reason in the next 10 years, the world's top 15 blockbusters, they are called a blockbuster. They are going off patent. And because of this off patent, there is a huge opportunity for a biotech companies to develop the biosimilars. And I was fortunate to work on a 13 biosimilar, and some of the biosimilars were world's the first biosimilar molecules, right? And they were ranging from a growth factors. Growth factors is considered to be a very simple one, to the complex monoclonal antibodies, MAPs. If might have heard the word MAPs, it's a monoclonal antibodies. Like all this rituximab, trastuzumab, they are called as a monoclonal antibodies. They are very complex to manufacture. So that is the reason there is a scope for developing biosimilar products. Now, even if you see, this is just one example. I have taken one disease, particularly called as psoriasis, which is a skin disease, and psoriatic arthritis. And here, if you see the pipeline of the products, except two products like apribilast and tofacitinib, all these are the biologics. So what does it indicate? It indicates that there is a trend towards developing a biologicals, right? And only these two, apribilast and tofacitinib, these are the chemical compounds, whereas the rest of these products, which I have given example like infliximab, etanercept, secukinumab, adalimumab, these are biological biosimilar products. So any immune related disease, if you see the pipeline is basically all biologicals, not a chemicals, right? That is the reason trend is shifting towards the biologicals. Even in case of a cancer, which is a very common one, breast cancer is the highest, followed by lung and prostate. And you will see that the products which are like a biological, these are called as a MAPs, monoclonal antibodies, trastuzumab, pertuzumab, these are called as monoclonal antibodies. This pembrolizumab, again, is a monoclonal antibodies. So these are all first line treatment for all the cancer. So I'm trying to focus that the treatment options are getting skewed towards the biologicals. And that is reason there are huge opportunity for developing biologicals and the biosimilar products. Now, what exactly mean by a biosimilar? Biosimilar means is a biological product which is a process by genetic engineering, which I talked to you, like recombinant DNA technology, and which is claimed to be similar to the biologicals. Biologicals means it's the originator molecule. And then a lot of other companies try to copy that. That becomes the biosimilar. So when it is called biosimilar, it has manufactured through the genetic engineering techniques like recombinant DNA technology, and which is found to be same in terms of safety, efficacy, and quality. These are the three important parameters. Those three important parameters should get matched to these biological products. And then it is called as a biosimilar products, similar to the biologics. So these are some of the examples like cytokines, growth factors, blood products like heparin, hormones like insulin, RSC, SCG, then growth factors like GCSF, erythropoietin, then monoclonal antibody. These are very focused one, like biosma, rituximab, trastuzumab. These are the example of biological biosimilars. Now, what is the difference? Biologicals or chemicals? As I said, the chemicals, they are very small in size. In terms of size, it could be like 10 to 1,000 Daltons, whereas biosimilars, they are very large in size. One dimensional is the characteristic of chemical, whereas biosimilars, they are very three-dimensional structure, and three-dimensional structure is very difficult to copy, correct? And the stability, chemicals are very stable. The molecules like aspirin, paracetamol, they are very simple to manufacture. They are very stable. Whereas the and then by using the genetic engineering techniques, they are manufactured. And biologicals or biosimilars, they are mostly protein in nature. That's the reason the allergic reaction are very common. That is not in case of chemicals like aspirin, paracetamol, atorvastatin. There are different terms used for biosimilars across the globe, but the meaning is same. The product which is having a similar efficacy, safety, and quality, they are called as a biosimilars. So say, for example, in India, it is called a similar biologicals. US FDA, it calls as a biosimilar. WHO, World Health Organization, they call a similar therapeutic product. Japan, it is called as a follow-on biologicals. Canada, it is called as subsequent entry. Subsequent entry means there was a biological already available in product, and the other manufacturer had copied it. That becomes subsequent biological. In Australia, it is called as a biosimilar. But the overall meaning is product which is having a similar safety, efficacy, and quality with that of the biological products. There are guidelines for a development of biosimilars, and the pharma biotech is highly regulated by guidelines. Even if you are outsourcing a raw material, there are specifications for outsourcing the raw material. It is a highly regulated one. And that is the reason there are guidelines coming from the regions, European Union, India, as well as US FDA. And these are all guidelines in terms of how to develop the biosimilar products. Now let us understand how this development of biosimilars happened. This is a historical development, and the Europe was way ahead of rest of the world to laying a path for a development of biosimilar. In fact, they started work in 2005. So the European Union, they have established a legal pathway, and that legal pathway has basically facilitated the development of biosimilars. And that's the reason the first biosimilar product was approved in 2006 in Europe. That product was Omnitrope. Then in 2009, WHO has published a guidance for biosimilars. India has its own guidelines established in 2012, which got updated in 2016. US was little late in terms of LING guidelines. So they started work in 2010, and then first guidance was published in 12, and the first biosimilar product which got approved in US was in 2014. And they have the first monoclonal antibody approved called as Remse in 2013. And there is the other products. But of late, US is the country which is very frequently approving all these biosimilar products. Now, what is difference between a biological? Biological means it's a first product. It is an innovator product. Biosimilar, it's like a generic product. Is there any difference in terms of the clinical development? Obviously, there is a difference. And here, when I talk about development, my focus is particularly on a clinical development. Clinical development means a phase 1 study, phase 2, and phase 3. So phase 1 is basically focusing on to study the profile of the product. How does it behave in the human body? So what would be its absorption, distribution, metabolism, excretion? And these studies, phase 1 studies, are all done primarily from a two perspective. One is to test the efficacy first time in humans. And the second is to characterize the pharmacokinetic profile. So that is the objective of phase 1 study, phase 1 clinical trial. So it is found to be safe and characterize the pharmacokinetics. It moves to a phase 2 study. Phase 2 study is a study which is called as a proof of concept study. And that proof of concept study is basically to establish its efficacy. Say, for example, you have hypothesis that certain compound has anti-diabetic property. So you have to really test whether it reduces blood sugar or not. So that is the reason that phase 2 study is done in a small patient groups, particularly to get efficacy as well as safety. That is called as a proof of concept study. And it is always in a small number of patients because you can't expose a large number of patients during early clinical development because there is always a risk of side effects. So once it is found to be safe and effective in a small number of patients, then it is moved to the large scale study that is called as a phase 3 study, which is also called as a confirmatory study, which is done in a thousands of patient, multicentric study, long-term study to confirm what was observed in a phase 2 study. So this biosimilar product, biological product has to go through the entire process. But whereas in biosimilar, it is accelerated pathway. Means you are just proving that it is a similar to biological products. So you not go through the phase 1, difference. So in case of biologicals, you have to do this, the characterization, functional characterization, the non-clinical study, phase 1 study, phase 2 study, as well as phase 3 study. Whereas in case of biosimilar, it is just to show that it is as good as that biological. So in that case, physical characterization is done, functional characterization is done, the non-clinical study is done, then PK, PD, and clinical study. So the extensive path is not followed by biosimilar because it is objective is just to show that it is similar to the biological products. Now, when you are developing a biosimilar product, it is always in comparison. So there has to be a reference product. Means when you are comparing its efficacy, there has to be a reference product, and that reference product used to be to authorize the reference product. It is always the originator product. So your product in development is compared with the originator products, and then one has to run full-scale safety, efficacy, and quality data in comparison with the biological products. Then pharmacokinetic studies are also done. In pharmacokinetic study, again, the PK characters like half-life, whether it is a linear pharmacokinetic, non-linear pharmacokinetic, all those studies are done. But these studies are always the studies done in healthy volunteers. Only in case of cancer drug, it is done in a patient because you can't expose the healthy volunteers to anti-cancer drugs, right? Otherwise, it is always in a healthy volunteers. And from there, the clinical studies are undertaken, which are called as a phase 3 study. There is no need to do a phase 2 study in case of similar biologicals. And I have explained to you why it is not required, because its main objective is to show that it is similar to biologic product. You are not going to establish any new thing, right? That is the reason phase 2 studies are not required. So you can directly go to the phase 3 study, which could be a parallel design. Parallel design means you have two groups. In one group, you have given a biological product, that is originator product. In other group, you have given the biosimilar products, and both the group are treated at the same time. That is called as a parallel, right? This row is parallel to that row. Crossover is first you give a treatment A, say for example, biologicals, and B gets the biosimilar products. And then after a certain period, you cross over that. The people who are sitting on this side get crossover to the other side. That is a crossover design. So both the study designs are applied to these studies. Can you move to next? Yeah. So even only with the pharmacodynamic. So what is the pharmacokinetic and pharmacodynamic? Anybody? Anybody who has some kind of a background of the pharmacology or drug? Pharmacokinetic is how drug behaves in the body. That is a pharmacokinetic, like absorption, distribution, metabolism, excretion. Pharmacodynamics means the actions of the drug. Those are called as the pharmacodynamics. So for pharmacodynamics, there are certain PD markers. PD markers like the pharmacodynamic markers. So if you are doing a treatment, the evaluating product for a cancer, so you can take the absolute neutrophil count as the PD parameters. So if it is found to reduce this absolute neutrophil count, that means this product is effective. So you do not test in the patient. In the same case, if you are trying to compare two insulin formulation, so you not test for the blood sugar level, but what you can do is you can do in glucose infusion clamp rate. Same way, the low molecular weight heparins. In that case, you also do a testing against clotting factors, anti-factor 10, anti-factor 2. Same is case for FSH, follicle stimulating hormone, which is used to induce the production of oocytes. So here, number of oocytes will be a parameter. So I was talking about the crossover comparative studies. So here you should do the comparative studies, and this comparative study could be equivalence studies, or it could be a non-inferior study based on the nature of your studies. In that case, you also do a testing against the clotting. Can I go to the next slide, please? Follicle stimulating hormone, which is used to induce the production of oocytes. So there number of oocytes. So there are different designs. One is called as equivalence design, other is superiority design, and third is like a non-inferiority. Equivalence means you are trying to show that it is equal. Superiority means you are trying to show that it is superior to the comparator. Non-inferiority is you are trying to show that it is a non-inferior. So you can adopt one of the designs. And the endpoints, effectiveness endpoints could be a disease specific. If you are trying to test it for the hypertension, so reduction in blood pressure would be the endpoint. Whereas in case of diabetes, it is a reduction in blood sugar level. So those endpoints can be selected based on the disease. Safety is very important. As I said, biosimilars or biological, these are the products which are of protein in nature. And because of protein in nature, they have all the potential to induce allergic reaction, which are called as immunogenic reactions. So that is, immunogenic studies are also required to be done. If you are trying to test it, once the confirmatory studies are done, in that case, then you get a marketing authorization. There could be possibility that even the confirmatory studies may not be required to be done. If product is found to be effective in the pharmacodynamic studies, in that case, even the phase 3 studies can be waived off. So when they are waived off, when there is a structural compatibility exercise, PKPD study has demonstrated that it is equal to similar biologicals, and there is a plan to do a post-marketing studies. In that case, even there is no need to do a phase 3 clinical studies. There could be possibility that I'm doing a study for one indication. Say, for example, it is rheumatoid arthritis. And then there is a possibility that it could be effective in ankylosing spondylitis, other rheumatic disease, or the psoriatic arthritis. In that case, I just do a study in one indication, and the data obtained in that one indication can be extrapolated to get approval for other indication. So the point here is, for all those indication, it is not required to do study indication wise. One study is adequate one. So that is called as extrapolation of data from one indication to multiple indication. So once you do all the studies, then absolutely the next thing is to get the commercial, the authorization or marketing authorization, approval from a regulator. And to get approval from regulators, there are 5 modules. And the 5 modules need to be submitted with all the details like administrative, legal information, summary from all those modules. So module 3 is a quality information. So the quality information is basically focusing on the lab data, chemical, pharmaceutical, and biological data. Then module 4 is a non-clinical study. These studies which are done in animals, they are called as a non-clinical studies. And the module 5 is a clinical information. which is the data obtained from phase 1, phase 2, and phase 3 studies. And basis this the product get approved by regulatory authority, and then it is ready to get commercialized. So to conclude overall, so there are a lot of challenges in terms of developing a biosimilars as well as biologicals, because it requires a huge infrastructure. The process of manufacturing the biological or biosimilars is very, very complex, very investment intensive. Despite that, there are a lot of opportunity because the 15 biologic products are going off patent in the next decade. And that provides a lot of opportunity. And I have spoken about the market, how huge is the market. 2.3 lakh crore is the market of biologicals. And that's reason there are huge opportunity for biotech and biopharma companies to take up the research in biosimilar development. And a lot of pharma company, they have shifted their focus from a traditional chemical product to exclusive biotech product. And there will be a lot of opportunity for professionals. Lot of opportunity means this is a knowledge intensive area. So one need to have the knowledge about chemical characterization, pharmaceutical the characterization, stability. So all those people from a background of the formulation development, people with the background of microbiology, who can characterize in terms of in vitro activities, plus people with preclinical the expertise as well as clinicals, they will have a lot of opportunity to get associated with biotech companies to be a part of the biosimilar and biological product development. With these are some of the resources. If you are interested, you can refer to these resources. And with this, I hope that they enjoyed this interaction. And I'm be happy to answer if there are any unanswered questions. Thank you very much.
Thank you very much for your extraordinary talk, sir, and for your rich words. It surely helped us to understand the marketing strategy and demand for the young minds. And sir is given very informative talk. Now, the topic is open for discussion. You can ask questions to sir. Sir is from industrial background, so you people is having good chance. Yes, sir, sure.
Not exactly the same. They're similar. And most of the interests we have on legal. One thing which was quite says functional character. So what are the yeah, very good question. In fact, it is out of my scope to talk about functional, but as I have been a part of multifunctional teams, I could understand that area. So your point is very correct. Any small change in the structure can make a huge difference. And in terms of biosimilars and biologicals, we call this as a residual uncertainty. You try to copy as that of biological. Despite doing all these efforts, there is always possibility that there is a residual uncertainty. Can't always 100% copy. And the process of development, it starts with a physical characterization. So you have to have the physical characterization, which is matching to the biology. That is the first important step. Moving from that, it's the biological characterization. There are bioassays, biological assays. Try to see that whether that compound, say for example, there is an anti-diabetic product, which is a biological molecule. And you have three models, bioassay models, wherein you can test its biological activity, whether it is effective in terms of reducing blood sugar or not. So through the biological assay, what you do is, yes, it is definitely reducing the blood sugar level. So it has functional characterization. We call it a functional characterization, which is based on the biological activities. So in the process, what you are trying to do is you are trying to minimize the residual uncertainties. You are trying to match the physically, chemically. If you are going ahead doing the biological or functional characterization, so in the process, you are trying to minimize the gap between biological and biosimilars. But and that is reason while developing biosimilars, focus is you can't compromise on physical technical. You can't compromise on the functional characterization. And then the confirmatory study will be required. And then ultimately you can see that it has to be effective in the patient. So this is sequential development. And I understand a small change makes a lot of difference. And even in biological, it is said that one batch manufactured is not same as that of second batch. There is a small change in manufacturing process. There are a huge difference. That's the reason there are stringent quality measures taken so that it matches with the biological process.
I understand the context of biologicals. It's easy to have a similar proposition. About small change in the functional. Basically, what was like saying that whatever the tests were done, the functional characteristic has to match. What is the scope to have this happening with the functional?
Right. There, as you go through the process, there will be always the gap reduction between the activities. That is, it will try to be similar to what you are expecting. And that is using this entire exercise. And this exercise is a comparative exercise. Even when you are testing a physical chemical characterization, it is complex the reference product. Functional characterization is the reference product. The clinical studies which are done in animals, they are also done in the comparison. And ultimately, the phase 3 studies are considered to be a confirmatory one. Beyond that, there is nothing to confirm, right? And that is even phase 3 studies are also recommended. So in the process, what you are trying to do is whether whatever gap is between your product and reference, it gets minimized to the extent possible. That is the reason this is called the residual uncertainty. You are trying to overcome that residual uncertainty. And this uncertainty is because of small change. Because you can't 100% copy to the reference product. I hope I clarified that aspect, right?
I just try to understand what is that gap is allowed. Like for example, in the function, standard or reference molecules.
Yes, it will be acceptable. Like I can give you an example. Bioequivalence studies. In terms of bioequivalence studies, the margin used to be 100 to 120%. On the lower side, 80 to 120%. So on lower side 80 and on the higher side 120. Right. So that margin is there because it is highly difficult to match 100%. That is the reason there is the margin. And even these studies which I talk, non-imperative studies, superative studies, that means there is a non-imperative margin, superative margin. Say when you are designing a study, which is the superative studies. So how much is that superative margin? Like 10 to 20%. Say this was case for a non-imperative. If it is 10 to 20% non-imperative, it's accepted. Right? So there are guidelines for it. Go, I'm clear about it. Yeah.
Thank you very much, sir. Sir,
Yeah.
Sir, we being from the universities and academics, based on our we develop some compounds, the Laxmikant. We analyze data in vitro studies up to certain level of the preclinical studies. Smaller. We have the barriers how to take it to the next level. Is there any procedure to reach to the doctors or the to perform the clinical? How to convince them what to approve or not approve? Is there any certain guidelines?
Yes, absolutely. So if you are pursuing on your own, take it to the clinical development. There are guidelines coming from organization called CDSO, Central Drug Control Office. And that authority is called the Drug Control India. So the subject of clinical trial getting permission lies with that authority. And there are guidelines for it. There is a regulation what are the drug and cosmetic acts. There is one chapter in that, clinical trials. So how to go about the clinical trials. So we need to do something in protocol. expert committee approves that product, and then you go to the FDA. Again, you have to provide the name of doctors who are going to be part of it. So my basic function is to get done all these user studies, but I do it for an organization. At the individual capacity, at a university level, you have to get collaborating with pharma biotech companies. So they have the expertise. They have all the resources. If it is done on your own through pharma company, you have to. It will be easier. Easier. It is not that you can't do on your own, but the path would be quite tedious path because you will have to identify all those resources required. Otherwise, pharma company, biotech company, they have all the know-hows. So it is their day-to-day work.
Nice talk. So, my query is, maybe antifungal vaccine. I'm asking because we have to vaccinate everything, right? As you said, right, in academic, we will not be able to take it to the further, like, so whether landmark or invest in some of such academics and anything which is interesting against something landmark would be the most important country to consider. First in India, we are number 2 in terms of dermatology. We have a spectrum of antifungal drugs, starting from metacolazole up to piclocol and all those molecules. So, the area of the fungal disease, we're looking at that possibility. So we have the process and we have a dedicated function, which is called as business development product portfolio management. So they always participate in a lot of conferences. They try to identify the research organizations who are developing this. So if I have anything, I can facilitate. Talk about bioactive molecules. Maybe it's not very stable outside. So why can't we use microbials not toxic? Yeah, there are very few vitamins like calcium or DHA or deficiency with the same molecule isolated from it. So that can be on the guidelines and it can make some new recipes some supplements. So yeah, it can definitely take it by that. I'm sure that's something. Okay, we have a question from this side.
Now understand the law of ingredients related to biosimilar and products. Wondering, sir, like our students, other skill sets if you work in landmark, the skill sets apart from some knowledge our students should have so that some intensive opportunity.
Right. I did speak at the end of my talk that those people who have a skill associated with the life cycle of the drug development. So drug development happens starting with hypothesis that certain compound which you have synthesized or obtained from a natural source and some kind of activities. So characterization of that substance, isolation of that substance, doing stability studies, then the spectrum of activities. So those skills will be all required. So ours is a company which involves from a basic research to the advanced research. Right? And basic research is particularly from chemical synthesis, and stability studies, and in vitro study, viral studies. Right? So particularly advanced development is I being a physician. So my role is particularly starts from a basic concepts, doing study first time taking it to the patient, and then into the team. So prior work always these skill sets are required. People about the those background having decency background, microbiology background, special interest in the research. That is also important. We have a dedicated center which focuses on the chemical entity research, and we have a center in Switzerland which focuses on new biology.
basic drug design. Now certain lead molecules which have been taken to certain stage and they were outsourced to other companies for further. However, those who fall into this value chain, they have all the role to play in a pharmaceutical.
Because at the end of the semester, I guess they have a project done. Maybe they'll come up with some biopeptides, for example.
They can collaborate with some of the pharma companies which are involved to the basic research and get a project which is important to them. I can give a simple example. I did my MBA in the pharmacology. When I did my dissertation at that time, but there was no train to take a project and your research work which is of interest. So I was the first student at that time. I got a project from a looking R&D. Bioactive is pretty on the hair product, which was combined with paparazzi. The hypothesis was paparazzi enhances bioactive. So they have your anti-tuberculosis product. They combine that anti-tuberculosis product with paparazzi. And the hypothesis was paparazzi enhances bioactive. I tested that and that was my so for them to collaborate with the early stage with the industry and they will be open.
Thank you very much, sir. Sir, we have quick questions on online mode. Can you please come over here?
The advantage of taking biosimilars. So first is
Sir, please. Sir, you please come. Yes.
So I think the advantage of biosimilar product is basically from a cost perspectives. Biological being innovator, they are highly costly products, and biosimilar medicines are like a generic products. So the cost effectiveness would be the advantage of taking a biosimilar medicines. And as far as the safety, efficacy, and quality, they are comparable with biologicals. So you get the same medicine with a less cost. That's the advantage. Okay. Any other question?
Two more questions are there.
Yeah.
Questions.
Please. Okay. Sorry. Okay. Question, specifically question. Okay. Okay. Fine. Fine. Thank you.
Thank you very much, sir. Thank you. I would like to request Professor T.A. Kadam, sir, to please felicitate Dr. Barkate, sir. Sir, please. Thank you very much, sir. Thank you. Sir is online. Sir online. Yes. Sir, Ale. Now, our next eminent speaker is Dr. Avinash Sonavne, sir. He is Professor, Department of Biosciences and Biomedical Engineering from Indian Institute of Technology, Indore, MP, and who is joining us with us virtually. So, I would like to request Professor T.A. Ganam, sir, to please introduce Dr. Avinash Sonavne, sir. Sir, please. Are you there, sir? Avinash Sonavne, sir.
Yes, yeah, I'm there.
Yes, thanks.
Welcome, sir.
Yeah.
I briefly introduce Professor Avinash Sonowne, sir. Actually, this conference was earlier planned in 2020. And being a advisory committee member of School of Life Sciences, Professor Avinash Sonowne, sir, have contributed a lot in first draft of the international conferences in 2020. He personally came here. Actually, he was introduced by late Professor Khobrade sir, myself, Khobrade sir, and Avinash sir, Sonowne sir, discuss regarding the conferences, and he has given promise. Along with him, he will call the experts from the outside the country also, but due to pandemic, it was not become possible. But still, sir has given the commitment, and now from Bhubaneswar, sir is giving the talk. Okay, so Professor Avinash Sonowne sir is presently in Department of Biosciences and Biomedical Engineering, Indian Institute of Science Technology, Indore, IIT. Presently, he is professor. Just meet Dean International Affair Outreach Program at Indore. Then he has many fellowships. Start from JSPS Fellow from Japan, Swedish Institute Fellow from Sweden, EMBO Fellow from Institute of Ilms, Germany, Swiss National Science Fellow, Zurich, Switzerland, EMBO Fellow from Sweden, and a Malit Fellow from Germany. Actually, sorry, this is a small typed one. I am finding difficult to read, but such a great personality is with us. So he is giving his presentation to this audience, which are joined both virtually as well as in offline mode. So, sir, please give your presentations, sir.
Let me share my slides first. Can you see my slides?
Yes, sir. It is visible.
It's visible?
Yes, sir.
Yeah. So, a very good morning to you all. So, first of all, I would like to thank the organizers and in particularly Professor Kadam for his. And Professor Kadam has mentioned I have been like associated with School of Life Sciences for quite some time, and it's really like an amazing experience to come over there, interact with students, faculty. Anyway, so today I'm going to talk about development of next generation drugs. As we all know that in last several decades, several drugs have been discovered, and many of drugs, there are still many other drugs they have been discontinued because of like several reasons, like this for the period of because of these evolutionary selection mechanisms. Many drugs they become ineffective. The microorganisms they develop resistance against many of these drugs. So there is a continuous challenge for development of these drugs, and that's why this concept is coming up that the development of this next generation drug. It means instead of developing a new drugs, so whatever drugs they have developed, we can do some engineering there. We can modify these drugs. This is this is also called as the drug repurposing. So because the development of these drugs, it takes and it's a very long process. Sometimes it takes 10 to 15, 20 years. So instead of developing these new drugs, can we make some changes in the existing drugs and make them more effective? So today I'm going to share with you my own experience of last 12 years. So our group has been working on development of one drug which is used for treatment of blood cancer. So I will just share my experience that what challenges we faced, how we developed, and where we are today. Before I start my talk, so since I'm interacting with many of you for the first time, so I wanted to give an overview about the research work going on in our laboratory. Our lab mainly works on two diseases, that is tuberculosis. In tuberculosis, we work on various areas, like we worked on many Mycobacterium tuberculosis, which is a positive agent of tuberculosis. We identify or characterize many new virulence factors and how these virulence factors, particularly the glycoproteins and glycolipids, because many of these proteins are present on the surface of the mycobacteria, and they help these pathogens to can you see the change in the slide? Are you able to see the changes in the slide?
Sir, is it possible to show full slide?
I think it's on the full slide only. I don't know why. Let me. So you're not able to see the full slides, right? That's what.
No, sir.
Actually, I have shared a full screen here. Okay, but you are able to see the content, right?
Yes, sir.
Yeah. So the slides are not moving on the screen. I can see that. Though I'm sharing this. Yeah. So, am I audible?
With it, because what happened when we are going for full slide, it is not going to be observed in online mode. It is not visible on online mode. So we can go with this. So if possible, you can send your PowerPoint presentation to us so that we can.
Oh, can I send my PowerPoint presentation? Can I send it now?
Yeah, it is visible.
Yeah, it's visible, but I have to change the slides. Okay, I think let's go, because otherwise sending and then that will take time.
Okay, no problem, sir. You go with that then.
Yeah, sure. So blood cancer, in TB, we work on various virulence factors, Mycobacterium virulence factor that helps in pathogenicity drug resistance, because it has been shown that this individuals, they get infected with tuberculosis and lungs and goes in the bone marrow. And this bone marrow is kind of a very safe environment for the TB, because the drugs, they are not able to penetrate into the bone marrow, and as a result, this bacteria survives and it causes chronic tuberculosis. And during this chronic tuberculosis, it leads to formation of the granuloma. So granuloma is nothing but a very solid mass formed in the lungs, and that leads to lung cancer. So we're trying to study the association of the chronic tuberculosis with lungs. And of course, we use various this animal models like zebrafish and mice for the in vivo study. The other area we are working on is the blood cancer, and this is what today I'm going to discuss about it. We are working on development of novel the asparaginase-based therapy, and currently this molecule is under clinical trial. So on the drug development part, we are working on development of these drugs for mainly two diseases, as I mentioned, anti-tuberculosis drugs, because there are several drugs are already available in the market, like isoniazids, pyrazinamide, ethambutanol. So these are called as the anti-TB drugs. But however, due to emergence of this multi-drug resistance bacteria, many of these drugs, they become ineffective. So we are trying to develop a hybrid molecule, small hybrid molecules, which can be used for the treatment of multi-drug resistant tuberculosis. And the anti-blood cancer molecules, this is what I'm going to discuss today. So, yeah. So we know that these immune cells, they are formed in the bone marrow, and the process is called as the hematopoiesis. So during hematopoiesis, the precursor cells which are present in the bone marrow, they differentiate into different lineages, and that leads to formation of various types of immune cells like red blood cells, white blood cells, platelets, granulocytes. Okay. So what happens in acute lymphatic leukemia? So we know that there are various types of blood cancers, like acute lymphatic leukemia, myeloid leukemia. Okay, so there are various types, but lymphatic leukemia is a major blood cancer diagnosed in many patients. So what happens in acute lymphatic leukemia? So there is a formation of when these cells, they develop into the bone marrow by a process called hematopoiesis. So there is formation of this malignant immature white blood cells. And these malignant immature white blood cells, because we know that these white blood cells, they are mainly responsible to fight the infections, because we get exposed to many different types of pathogens. And these white blood cells, they help us to fight against this infection. But when there is formation of this immature white blood cells, these cells, they are not able to fight against the infection. And when they turn into a malignant, that is the cancerous cells, these cancerous cells, they develop very rapidly. And as a result, what happens after some time, these malignant immature white blood cells, they continuously multiply, their number increases. And when their number increases, the number of healthy white blood cells decreases drastically. And the bone marrow becomes just overcrowded with these cancerous cells. And that leads to blood cancer. And as I mentioned, various types of cancer, but this acute lymphatic leukemia, it represents 23% of all cancers diagnosed in children. And if you look at the number of patients, the patients are gradually increasing in India. Few years back, roughly 15 to 20,000 patients were diagnosed per year, new patients. But now, as per this recent data, more than 35,000 new ALL cases that are diagnosed every year. And in global scenario, there are more than 900,000 people that are affected with ALL. So, yeah. So there are various phases of cancer cancer chemotherapy. There are, it is classified, there is no, I mean, it's a low number of these cancer cells are present. In high risk patients, ALL patients, the number of cancer, it's an advanced stage of the cancer. In standard these ALL patients, there are 3 phases of chemotherapy. They are such as induction, consolidation, and maintenance. So in induction, the combination of chemotherapeutic drugs are given to eliminate or to kill the rapidly dividing cells. And as you can see, in all induction, consolidation, and maintenance phase, the asparaginase molecule is used. So there is no substitute for asparaginase drug. Even in high-risk ALL patient, the asparaginase is used. So what I try to say that the asparaginase is a very important component of blood cancer chemotherapy, and there is no substitute for asparaginase. And various studies have shown that when this asparaginase is used in the chemotherapy, the survival of these blood cancer patients, it increases to more than 80%. Okay. So how these asparaginase works? Yeah. So how this asparaginase works? So this asparaginase enzyme is convert this asparagine into aspartate. In normal cells, what happens? This aspartate and asparagine, these are 2 amino acids which are present in the blood. So our healthy cells, what they do, they take this aspartate from the blood. Okay, and when this aspartate is taken up by this our cells, there is an enzyme called as asparagine synthetase enzyme. It converts this aspartate into asparagine. Okay, and then this asparagine is used for the protein synthesis. Okay, and then cells divide. Another source is the asparagine is also present in the blood. So these cells, they take up this asparagine directly from the blood and use this asparagine for protein synthesis. But what happens when these healthy cells get transformed into the cancer cells? This cancerous cells, they don't express this asparagine synthetase enzyme. Okay, so they don't, these cells Hello. Yeah, can you hear me?
Visible.
Can you
This.
hear me?
Yes, sir.
Well, there is some network issues. Can you see slides now?
Yes, sir.
Yeah, so I'll just quickly, because we already.
Sir, can you minimize the slide content so that front side is somewhat bigger?
Is it okay?
No, sir, it is minimizing. Sir, go for slideshow.
Yeah, I tried that, but I don't know what is happening.
I think it is okay. Jonas sir, please go to share and then share the screen so it will be visible.
Is it visible now?
Yes, sir.
Sir, go
So
for
currently there are
slideshow and then share it on Zoom.
Are you able to see slides now?
Okay, sir. Okay, sir. Go for it. Maybe some problem is there.
Yeah, I don't know why this is happening. So there are various drugs, asparaginase drugs are currently used in the market. However, these drugs, they have several limitations, like these drugs, they have very low serum stability. That is, when it is given to the cancer patients, these drugs, they are rapidly degraded, and so that the patient needs to take repeated doses of this drug, and that causes toxicity to various organs. Another drawback of this asparaginase drug is that it has high glutaminase activity, and which leads to neurotoxicity because of this high glutaminase activity. And since this drug is derived from bacteria and given to human, the human body recognizes this drug as a foreign molecule because of its immunogenic nature. And these patients, they produce antibodies against these drugs. And these antibodies, they neutralize these drugs. And as a result, its therapeutic efficacy goes down. But there are this PEG asparaginase molecule is available in the drug, but its treatment cost is very, very high. Okay, as you can see, the treatment duration is about 28 to 30 weeks, and the total cost goes up to 130,000 US dollar. And because of this various undesired side activities, this asparaginase drug, they cause hypersensitivity, frequent infection, joint pain, swelling of various organs in the patients. Another major drawback of this drug is that, as I've said, that it produces antibodies in the ALL patient. So when it is given, this drug, asparaginase drug, is given to ALL patient, the administration, it causes a formation of anti-asparaginase drug. And when there is relapse or recurrence of this cancer, and when this drug is given second time, so these patients, they already have antibodies. Okay, and so when this drug is given again, these pre-existing antibodies, they bind to this enzyme and neutralizes. So the success of the recurrent cancer, blood cancer, is very, very less because of the presence of these antibodies in the patient bodies. So then, what is the solution for to overcome these challenges? Is to develop a new drug which has better stability, which has less glutaminase activity, which will not cause any neurotoxicity in the patients. This drug should not have any immunogenicity, because then there won't be any antibody production in the patients, and so that we can improve the treatment of relapse cancer patients. So to overcome these challenges, in last 12 years, what our group is working on is to develop a new asparaginase molecule. So we use a protein engineering approach where we modified some amino acid. You know that there are some B cell, T cell epitopes are present in the protein, and some of these epitopes, they are very immunogenic in nature. Okay. So we modified some amino acids in the B and T cell epitopes and to reduce its immunogenicity. So by using this site-directed mutagenesis, we mutated many amino acids which are present in the B and T cell epitope. And through this protein engineering approach, we created more than 12,000 mutants. Okay, and then we screen all these mutants and we check which mutants they have more asparaginase activity, low glutaminase activity, which reduces the neurotoxicity, which mutants they have low immunogenicity. So we screen all these 12,000 mutants, and finally we found that there are 4 mutants which has these desired properties. So these 4 mutants, we found that one mutant has high asparaginase activity, another mutant has low glutaminase activity, one of these mutants, they had low immunogenicity, and it does not bind to pre-existing antibodies. And we found that among these 4 mutants, one mutant, it was specifically killing more leukemic cells without affecting the normal cells. So we have 4 different mutants with these properties. So we identified that what are the changes are happening in there in this mutant, which are giving these properties. So we identified that by whole genomic sequencing, and then we introduced all the changes in one mutant. So now finally we have one asparaginase molecule. We named this molecule as M-aspar, and this single M-aspar molecule has all these properties now. It has more enzyme activity, it has low glutaminase activity, it is low immunogenic in nature, it does not bind to the existing antibody. And this is what I'm going to show in next this couple of slides. So, so we purified these mutants, and as you can see that this M-aspar molecule, which is a single molecule, this asparaginase drug now, which has all desired properties which we are looking for. And we found that this M-aspar molecule has high enzymatic activity. As you can see, the catalytic activity is high. This mutant did not show any glutaminase activity. As I said, this glutaminase activity is responsible for the neurotoxicity. So this low glutaminase activity in this mutant will not cause any neurotoxicity in the patients. So similarly, as I said that this asparaginase molecule is highly immunogenic in nature, and it produces antibodies when it is given to the patients. So we checked immunization, or we checked immunogenicity of this asparaginase molecule. So what we did, we took this mice, and in one set of mice, we injected a normal asparaginase drug, and in another set of mice, our M-aspar drug, which we had developed in our laboratory. So we gave 3 booster doses. First dose was given on day 0. Second dose was given after 21 days. Third dose was given after 45 days. And after 66 days, we collected this serum from this immunized mice, and then we checked antibody production in mice. So what we observed that the mice which were given this M-aspar drug, which is developed in our laboratory, there was very less IgG and IgM antibody production in the mice, indicating that this M aspar molecule is significantly less immunogenic. It does not produce any antibodies in the patient. So as I said, there is another drawback is that when there is a relapse blood cancer, because of this presence of, and if these patients, they have taken this asparaginase therapy, so these patients, they have antibodies in their body. So when this asparaginase molecule is given again, so these pre-existing antibodies, they bind to this asparaginase molecule and neutralizes, so that its therapeutic efficacy is hello. Hello, can you hear me?
Yes, sir.
Sir, go ahead, sir. You're visible. Your sites are also visible. Sir.
Sorry, there is some connectivity issue here. Anyway, I think so. I will. Okay. So
You can proceed, sir.
I just so what
is here is Professor Giri sir is present in audience. He want to talk some point regarding your presentation. Please, sir. Professor Giri sir from NCL.
Yeah. Okay. I mean, good morning. Still, I think we are in the good morning session. So I am doing something like a SOS job. So I also do not know much, but what I understand from his lecture and probably what he was supposed to show further is that with all this site-directed mutagenesis and so on, they have developed a battery of asparaginases, which have important, I will say, role in the drug discovery against cancer. And he come up with 4 molecules, 4 asparagines, which are probably modified by this site-directed mutagenesis coupled with artificial intelligence and machine learning probably. And then one of the molecule, which one of the recombinant enzyme, which is asparaginase, showing a I will say desired properties as well as some advantage to existing asparaginase, and probably he would like to take it further. So as I think in earlier talk, you got idea that how one has to check the activities of the molecule which is newly developed with a reference molecule. Since the asparaginase is already known, and it is already used as a drug molecule, so the asparaginase that he has developed has probably a superior quality, and then less side effects. That's what I think the presentation was going on. So probably his further presentation will was proposed to be giving like more ideas like how this molecule is superior in several functionalities, like functionalities with I mean their in vitro characterization as well as with probably animal trials, as well as I do not know whether he has conducted the preclinical trials or maybe he is in the process of applying for the DCGI approval, as we have mentioned, because anything that has to grow as a drug, there are a lot of regulation and it has to go with that. So I think let us conclude about it. I may not be in a position to answer the question about like what are the further developments in this respect, but if there is any question related to the part that he showed, I will try my level best answer with the help of the senior faculties here. So if you want to ask any questions, particularly from student side, we'll see whether anybody of us can understand or answer this question. Is it okay if all senior people agree? So any question from students related this? Okay. So there are no questions. I think we can conclude then. Thank you.
Really, thank you very much for Giri sir. And I also thankful for Dr. Avinash Sonone sir for delivering his talk on next generation development of next generation drugs, challenges and strategies. But I really apologize for the technical issue
and which was tackled by Dr. Giri sir. So thank you very much, sir, and thank you, Avinash sir. Sir has very nicely presented on the role of asparaginases in cancer, and also role of protein engineering, site-directed mutagenesis, and all. These terms are routine for the students, but they really don't know the exact applications of these terms. So after your lectures, they will learn from something regarding these terms and all, sir. So thank you very much, sir. Now I would like to request everyone for high tea. So tea is waiting for you, and we will sharp resume over here at 12. There is again online lecture, so you should be here at sharp 12. So enjoy high tea.
बस तू भाग मिल्खा, ओ बस तू भाग मिल्खा, भाग मिल्खा, भाग भाग।
Music का डोज, मस्ती हर रोज। Get ad-free music and unlimited downloads with Hungama on the V app. Download now.
जगातील सर्वात मोठा लॉकडाऊन भारत राबवतोय. कोरोनाच्या जागतिक महामारीमध्ये कोट्यावधी लोक अडकलेत. स्वातंत्र्यानंतरच्या सर्वात मोठ्या स्थलांतराला आपल्याला सामोरं जावं लागतंय. आम्ही जगतोय. आपल्या जगण्याचे वागण्या याच कोरोनावर मात करण्यासाठी पोलीस रस्त्यावर, डॉक्टर्स हॉस्पिटलमध्ये आणि शास्त्रज्ञ प्रयोगशाळांमध्ये कोरोना योद्धा बनून अहोरात्र काम करतायत. परंतु या अदृश्य स्थितीतील कोरोनाला आटोक्यात आणायचं असेल तर ट्रेसिंग, टेस्टिंग आणि ट्रीटमेंट या मॉडेलचा प्रभावी वापर हा एकमेव उपाय सध्या समोर दिसतोय.
लातूर असेल, परभणी असेल किंवा हिंगोली असेल, तसेच नांदेडचे सॅम्पल्स हे औरंगाबाद किंवा हैदराबाद इकडे पाठवण्यात येत होते. त्यासाठी वेळ पण जात होता आणि त्यासाठी पैसा पण जात होता. सोसायटीमध्ये मोठ्या प्रमाणामध्ये पॅनिक पण त्या काळामध्ये झालेलं होतं. अशा परिस्थितीमध्ये विद्यापीठाने सुद्धा या मोहिमेमध्ये यामध्ये भाग घेतला पाहिजे. हा सामाजिक दृष्टिकोन समोर ठेवून विद्यापीठामध्ये जी उपलब्ध साधनसामुग्री आहे, उपलब्ध मनुष्यबळ आहे, त्यांच्या मदतीने आपण या प्रकारची सुविधा विद्यापीठामध्ये उपलब्ध करून द्यावी, हा त्यामागचा मुख्य उद्देश होता.
सॅम्पल टेस्टिंग काही विद्यापीठांचं मॅन्डेट नाही आहे, पण मी असा विचार केला की जर विद्यापीठाकडे इन्फ्रास्ट्रक्चर आहे आणि जर आता त्या इन्फ्रास्ट्रक्चरची समाजाला गरज आहे, तुमची एक्सपर्टीज आहे, मग त्या एक्सपर्टीजची समाजाला गरज आहे, कारण ती एक युद्धजन्य परिस्थिती होती आणि युद्धजन्य परिस्थितीमध्ये प्रत्येकाने आपापल्या क्षमतेनुसार त्या युद्धात सहभागी व्हायला हवं असं म्हटलं जातं. आणि म्हणून मग आम्ही ठरवलं की आपण या युद्धात आलो पाहिजे.
कोविड-19ची टेस्टिंग लॅब सुरू करण्यासाठी 2 गोष्टींची आवश्यकता होती. एक म्हणजे ICMRची मान्यता आणि दुसरं म्हणजे NABLचे ॲक्रेडिटेशन. कुलगुरू डॉक्टर उद्धव भोसले, जिल्हाधिकारी डॉक्टर विपिन इटनकर, लॅब संचालक डॉक्टर गजानन झोरे व स्थानिक लोकप्रतिनिधींच्या प्रयत्नांना अखेर यश आलं आणि स्वामी रामानंद तीर्थ मराठवाडा विद्यापीठ हे विक्रमी वेळेत NABLचं ॲक्रेडिटेशन मिळवणारं भारतातील पहिलं अकृषी विद्यापीठ ठरलं. टेस्टिंग लॅबसाठी उपलब्ध असणाऱ्या यंत्रसामग्री व्यतिरिक्त अजून काही यंत्रसामग्रीची आवश्यकता होती. ती खरेदी करण्यासाठी नांदेड, हिंगोली आणि परभणीच्या जिल्हाधिकाऱ्यांनी निधी उपलब्ध करून दिला.
डेलीचे 300 ते 500 पर्यंत सॅम्पल रोज होत होते आणि त्या लॅब इस्टॅब्लिश झाल्यामुळे नांदेडलाच नव्हे तर लगतच्या परभणी आणि हिंगोलीचे पण आम्ही डेलीचे 100-200 सॅम्पल आम्ही रोज करून त्या जिल्ह्यांना सुद्धा इथे मदत झाली. त्यामुळे ही लॅब हे इस्टॅब्लिश हेही महत्त्वपूर्ण आहे की त्यावेळेस कोरोना हा पीकवर होता तेव्हा डेलीचे आमचे 500 ते 800 सॅम्पल दररोजचे यायचे. त्यामुळे ही लॅब तेव्हा शॉर्ट रेकॉर्डमध्ये आणि याची दखल गव्हर्नमेंट ऑफ महाराष्ट्राने पण घेतली आहे की मराठवाड्यामध्ये आपली दुसरीच लॅब होती, एक औरंगाबादला आणि दुसरी नांदेडला तयार होऊन या लॅबमुळे जवळपासच्या 3-4 जिल्ह्यांना सुद्धा ज्या मदत झाली.
जैवशास्त्र संकुलातील होतकरू विद्यार्थ्यांनी स्वेच्छेने कोविड टेस्टिंग लॅबमध्ये प्रशिक्षण घेऊन कामाला सुरुवात केली. मर्यादित मनुष्यबळ, रिसोर्सेसची कमतरता, टेस्टच्या संख्येत व पॉझिटिव्ह टेस्टमध्ये झालेली मोठी वाढ यामुळे सर्वांमध्ये एक प्रकारची भीती आणि ताण जाणवत होता. परंतु यावरही मार्ग काढत कोरोना विरुद्धचं युद्ध त्यांनी सुरूच ठेवलं.
तर मला खरंच त्या सावित्रींच्या लेखींचा, आमच्या विद्यार्थिनींचा अभिमान वाटतो, विद्यार्थ्यांचा अभिमान वाटतो की त्यांनी एवढ्या याच्यामध्ये आणि काय म्हणतात त्याला रिस्क असताना सुद्धा काम केलं आणि त्यांच्यामुळे ॲक्च्युली ही जी लॅबचं जे काय टेस्टिंग सुरू झालं हे खरं तर त्यांनी दिलेल्या सपोर्टमुळे झालं. कारण त्यावेळेस बाहेर माणसंसुद्धा भेटत नव्हती, एक्सपर्टसुद्धा, मॅनपॉवरसुद्धा अवेलेबल नव्हती.
ॲडव्हान्स स्टेट ऑफ आर्ट इक्विपमेंट्स आम्ही ह्या ठिकाणी आणू. त्याने त्या इक्विपमेंट्सच्या वापरानुसार जे काही विविध प्रकारचे वेगवेगळ्या प्रकारचे जरी व्हायरसेस आले त्या
Please keep quiet. I would like to welcome all, and especially I would like to welcome Dr. Kuntal Chatterjee sir, who is Vice President of Pharma Circuit from Berlin, Germany, and who has joined us virtually. So welcome sir. I would like to request Dr. Hemalata Bhosle ma'am to please introduce Dr. Kuntal Chatterjee sir. Madam, please.
Good morning to one and all. Kuntal, can you hear me?
Yes, I can hear you well.
Okay.
Thank you.
So I am here feeling proud and privileged to introduce very dynamic person, Mr. Kuntal Chatterjee, who is at present working as BD consultant, that is business developer consultant, and vice president of Pharma Circuit company, which is a pharma-based company situated in Berlin or Frankfurt, where headquarter is there in the Frankfurt, Germany. I'm very glad to introduce him because not only he is working as a business accelerator, speaker, and futurist, he is also a freelancer and advisor for various pharma companies globally. After his master's in MSc Microbiology from Government Institute of Science, Aurangabad, he thought out of the track, means instead of writing for CSR exams or joining any pharma industry as a QC or RD, what he did MBA in pharma management and data analysis from SVKM's Narsi Monji Institute of Management Studies, Mumbai. He is now assisting companies globally to grow their business and also helping them with licensing needs, business growth, as well as M&A activities. He is also business owner with experience in global business development, consulting, market access support for Germany and Europe. With this brief introduction, my heartful welcome to Kuntal Chatterjee, and I request him to start his talk.
Great. Thank you, Hemaji, for such a great introduction. I'm really privileged and grateful to you all. And greetings from Berlin. So I have started my education in Aurangabad from Government College, GASCA, and then Government Institute of Science, and then of course Narsi Monji. So I owe a lot to, Maharashtra, India, and thank you for that. I'm originally from Bengal, and then, I came to Maharashtra and did my studies there, and then, pharmaceutical career started. Of course, I started in quality control. I am just trying to refresh for the, for the students who are our future. So I started in quality, and then I moved gradually to regulatory affairs. And that is the reason, I will show you today how to win with new drugs, what are the, let's say, success rate, how to go ahead, in with your dream, where you can position yourself in the industry. So, having started in quality control, I learned the basics of the testing, and of course, I started with Dr. Reddy's labs in Hyderabad. So from Aurangabad, I moved to Hyderabad. And there, after spending few years in quality control, I graduated to regulatory affairs. And specifically during that time, I went to US in 2005, where I was submitting dossiers, to the US FDA. And as we go through the presentation, you can see that how, we are, let's say, positioning our drugs, and, how we are really, taking that to approval. So currently I am doing business development, and but my core expertise is technical, and, I am doing this for around 13 years time. Somehow my screen share is not allowing me to, take it to the new page. So I will stop share and I will share again. Please bear with me for 1 second. 1 second, we'll do that again. Okay, so now I think it's working. Let me share that again. Sorry for the inconvenience. I hope it will work. Okay, so somehow the screen share is not allowing me to share and move on to the next presentation. So what I will do is, I will just go ahead with, with the PowerPoint, and without, going to the full screen. So as you can see, friends, in the next slide, it is all about, making your drug, to the international community. Now, I'm sure, many of you must be aware of this process. Whenever you wish to have, your new research, or, your idea, or the INN, or, something like which is your brainchild, when you want to take it to the commercial, you need to really do some, market research. So of course you are developing your compound, maybe it's for pain management, maybe it's for COVID or influenza, anything, you wish to develop. You first have to analyze yourself, what are the target conditions and available treatments currently in the market. And you can get that easily, I mean, you have Dr. Google, you have various publications. So first is to really find out whether your research is really adequate and, is really, let's say, forward looking. Of course, the guidelines are established, so you don't need to worry. You just, I have given you all the details about the scope and all that, in the, let's say, counter below. You can go through fda.gov or various other sites. You can also ask me for consultation. So the process is, you look at the target conditions, you look at the available treatments, and then of course you have to do, pilot preclinical. Like nobody will allow you to really go and test it to a human population. So first is preclinical, and once you get a data based on your laboratory, results, you do preclinical, then you have to assess the benefits. And of course, this has all to be done in conjunction with the, health authorities. In India, you have this drug controller in every state. You have in, drug controller. You also have DCGI in New Delhi. So you have to have different form of applications, and basically you have to find out with these regulatory bodies, starting with India. And of course, you have to, of course, file your own patents. This is needless to say. So you have to follow specific guidelines, and then when you are really sure that you have a unique INN, which is like a drug compound or a biologics, then you can really put forth your application to Indian FDA, US FDA, or any FDA, Food and Drug Administration of your choice. and then you can go for market access. So typically, if you see the patent, you get for around 20 years, but by the time the patent is granted, you have around 16, 17 years left. And then, of course, you have to manage the, approval timing. And that is the reason, you can see on the screen that it is very important to have an accelerated approval if there is a promising therapy. One example is this COVID-19 vaccine. We saw that, in 2020, this virus really caused a lot of, damage to the world, but nobody thought that a vaccine will be approved, in 6 to 8 months. And it was done less than 6 months. So I think this concept was applied because already, there was a platform, mRNA technology was there, and people were at an advanced stage for Ebola vaccine. And, they were very, let's say, confident that, they can use this to the benefit, and they got fast track just because, let's say, the managed strategies for managing risk was eminent for US FDA and all other regulations. And that is how, you got vaccine approval in a record time, which was never possible in the past. So in terms of drug development designations, clearly there are 2 or 3 pathways available. Usually, priority review is given for promising drug candidates. That means, for the molecules where instead of 10 months or 1 year, approval period, you can get approval within 6 months. There are also, certain designations like breakthrough therapy, like we had, for COVID. We can have fast track for certain, drugs. We started this all discussion, I think, in 1992, when there was huge hue and cry in US for the approval for the HIV, drugs. So that's where all these things started. And now there is an established process. And also, everybody is helping to reduce the clinical burden. Usually when you have 2, 3 pathways. For new drugs, you do a new application, like, it's a new 505, a new one. It could be NDA or it could be a biologic application, depending on whether it's a chemical or biologics. For the generics, I mean, you all must know that India is one of the major players in generic industry, and it controls the world with its generic products. So for all the generics, there is a reduced pathway where full-blown clinical trials are not required. So that means when innovator starts with the new molecule, or if you invent a new molecule, we have to do clinical trials using all, possible, patient population, and it could be on a tune of from 1,000 patients to, around 10,000 patients. But when you launch generics, that means, the quality is same to the innovator, and you can do the studies from 12 to, around 100 patients. So the cost is limited, and also the market access is better because of the huge production scale, and India is leading that. So without waiting for further ado, you can ask me all the questions, now or later on, as you prefer. So there is a unique, opportunity for all of you who are graduating now, later, after a few years, to choose your career wisely. So either you can become an R&D scientist, or, you can go for business development like I am doing now after spending a few years in quality and regulatory, or you can simply be part of any intellectual property department. You can look at what are the drugs coming onto the market. And with that, I start how to position, business development. So always, if you see all the pharmaceutical companies, they are always struggling in finding the right product or portfolio mix. Because unless you have the right product with the right mix, not only it's a problem for market access, but it's also a problem of overburdening the industry with too many approvals. So this is the most important thing for all the scientists, for all the pharmaceutical company, to have the right kind of business development strategy and vision. And in general, what I do now, I'm shifting focus. So I explained to you about new drug approval, how the process is. Now I go into details, in terms of business development, it's, of course, all the companies wish to manufacture, wish to do their own innovation and do everything in-house. But doing everything in-house is practically impossible. As you have seen that, one of the biggest pharmaceutical company, Pfizer, was not able to do vaccine itself. So it has to take help from BioNTech, which is a small, company located in Germany. So they did some kind of, collaboration together, and that's how they got the first vaccine. And also, if you see on many other companies, they collaborate very closely with universities, they work with students, professors, because it's simply not possible to do everything in-house. And that is the reason, business development is such an important thing in this real world. So what we do is, my job or, our job, all the business developers around the world, we actually have, we attend a lot of conferences, we meet a lot of new partners, we travel a lot. And in that process, we establish, relationships. In the process of last 15 years, I have developed over 22,000 relationships, in the pharmaceutical world. I also have a good follower in LinkedIn. So what we do is we capture those relationships. We know exactly what are the things, which are available for market access, and our role, business developer role, it's not me or you, it is us. We take this product from lab to commercial. And for that, of course, a lot of networking. It's all, modern world, it has become better, after COVID, because people are now just sitting at home using Teams, Google Meet or Zoom, they're networking. So you have to network, you have to really see and connect the dots, because, sometimes you find the opportunity lying somewhere else and not in the country or region where you are located. So that's the first part of the process, where we list down potential partners. Then, of course, before sharing any of your know-how, you have to have an NDA, non-disclosure agreement, or a CDA in place. Without having an NDA or CDA, you should not share your confidential data, because this can be, wrongfully used by somebody, and they will simply take away all your innovation. So very important part of the process before you really become, fully integrated or engrossed in the system, you have to sign a non-disclosure or a confidentiality agreement, as we say, with your partners before disclosing anything which is secret, which belongs to you. And then once that NDA is signed, you start the process, you start providing your documents, the marketing partner. So we have two partners. One partner is the development partner who develops the product. It could be university, it could be a lab, it could be anything, and the other part of the puzzle is marketing partner. So marketing partner usually takes the product to the market. Development partner develops the product. So there are two puzzles, and in between, there are business developers like me who helps to connect the dots between the development partner, which is a research lab, and the marketing partner, which is taking the product to the end customer. So, and to make it more interesting, there has to be a business case. Now, in simple terms, business case just have 2-3 unique things. So we have to just do a rough forecast on what is the actual potential. For example, what is the number of patients that we are going to target, and what is the price point we are going to, arrive there. So there is a price volume relationship. Once we know what is the volume, that means the target patients. For example, in COVID, we knew that there are millions of patients who can benefit, but we were not sure on what price we will keep. So always this price volume relationship is quite important for anything in pharma world and everywhere else. So this has to be rightly discussed, if required, also with government agencies, because in the process, there is an established process of getting this market access, and it has to be informed well in advance. So once all that is done, once we know exactly, let's say the time to market and the price and product appearance. So I'm just not going into all the details, but if you see in this slide, it is quite important to remain focused. I hope I am with time. Yeah, I still have 14 minutes. So most important is to see what will be the time to market. You have a good product, but it will take a lot of years to actually prove that it is safe. So unless your product is safe and unless you can prove that, the preclinical studies done on, like mainly animal studies is proving safety, and then you have to prove efficacy, you cannot really take it to the next step. So we have to always think by making the lab work in such a way that time to market is clear. And then, for the product, there are few things which we need to take into consideration. Now, after the COVID crisis, we got a huge burden on the supply chain. That means, all the containers, they were mostly, let's say delayed. They got stuck in various ports. There were a lot of restrictions. And in a way, due to this supply chain constraint, a simple cargo from India to Europe or India to US or China or, anywhere else, Turkey, it got delayed. And not only the time delay happened, there was also delay in, manufacturing. So nowadays, the global leaders are thinking of really cutting short this, issues with, travel and logistics. And they are trying to have at least local packaging operations, because we have seen that finished product also have to be packaged, and there are a lot of requirements on, serialization. That means, to avoid counterfeit and, fake products. There are a lot of requirements in pharmaceutical. To mitigate all this risk, they are trying to have hubs in different territories. And as you might know, for many countries, it is mandatory to have retesting on arrival. So whenever a drug comes from India, China, or anywhere, it has to be retested. And this retesting also, delays the whole process of launch. So in a snapshot, if we see, not only the quality is important in the lab, you have to monitor strictly the quality of the product. We usually, when we develop the product, we put it under room temperature, we put it under accelerated temperature, because the product, can travel over Gulf, over territories where temperature is extreme. So this is called stability studies. We do stability studies and we find out that the quality of the drug, which is written in every pack, you see it is mentioned, 2 years or 3 years, from the date of purchase. That means in lab, the product is already tested for such a time. That is the reason you have for every pharmaceutical product an expiry date. And you should always throw the product if it's, over expiry date, or you should retest it before consuming, because that is what is, done in the lab. They test the product, the efficacy, how much content is still left. And in this, whole part of the game, I will always refer to time to market, because, without a proper time of launch, because usually you have a rush, whenever you have a launch is generally dependent on patent expiry for the generic product. So if you are not there on day one, then your market share sinks. And also there are various methodologies available. For example, in US, if you are first to launch, then you can get 180 days or 6 months of exclusivity. That is as good as, you being alone in the market. So this is the right reason to have, right product in right time and with the right price. Now, having, let's say covered all the topics on business development, I'm open for all the questions. I will go shortly about what are the, what is the real knack of how we can really grow. So it is all about relationships. You should not care about money or anything else. Position, money, everything will come. I mean, this is as good as I say. If you really build relationships, and really think that this can be done, everything is possible. And I will share the slide deck. You can, go through it. And of course, with relationship, communication. I mean, this is very, very relevant for pharma industry. And you have seen the communication flow during the COVID times. There was a lot of bad communication, fake news, but there was also good communication, which was helping the whole industry in general. In terms of over-the-counter products, what I was explaining for last 15-20 minutes, I think I have 9 minutes time now, was mainly prescription products, which has to be prescribed by doctors. But there is also other products, which are called OTC or over-the-counter products, which needs no prescription. And now, thanks to Dr. Google, we all know what we should eat and drink. I mean, my faith is that in next 10 years' time, we will exactly eat and exactly drink and exactly do such exercise that we will be, less dependent on all the medicines except this COVID and all that. So very rapidly, this over-the-counter medicines, these food supplements are really picking up. And I can see that this trend is also one thing where you guys can think of. You can really think of developing something which has less regulations of clinical trials. You can launch within 1 year, for example. You don't need to wait for 7 to 10 years like drugs. And here, what the simple, example is to really look at every country in the world have a limit. For example, if you build a protocol like, you have to take vitamin D. You develop a vitamin D drug, but there is an instance where vitamin D cannot be absorbed fully if you don't take vitamin C. So how to titrate that, how to do the OTC stuff, and of course, with the right limit, which is available per country, there is a register. You can look at it and you can actually benefit from having, from day one, not crossing the limit. And this drug doesn't require any clinical trials, because these are all based on established use. That means, these are like our Ayurveda. It has established, it has been, a proof of established use. So this is quite, fast. People tell me that, there is no benefit of OTC. But if you see even, small things like ginger, haldi or curcumin, this has huge potential in the Western world and everywhere. And this has really now picked up during the COVID times. Immunity is the key, and hence this segment is growing, quite a lot. Now, last few minutes, I want to tell you about Azeta Pharma. So this is a company, which we started, of course, my wife, who was also studying with me, in Aurangabad. So we both started the company along with a German friend of ours, Mario Schaefer. And we thought that, we should start something which is more forward-looking. And this company is forward-looking because, we are doing free seminars. Your next seminar is on 20th of May. You are free to attend that. There's no fees for such seminars. So what we are doing is, through this company, we are having a platform which is called Pharma Circuit. And with that, we are promoting, a lot of BD projects, ideas. We are giving free seminars. And we are also doing some kind of mentoring. Across the world, wherever there is, help required. In some cases, it is free of charge. Some cases, whoever can pay, we are getting, paid. But most importantly, this, Pharma Circuit platform brings academics, brings, commercial guys, and also brings all the intellectuals, in one platform. And of course, there is no, let's say, right or wrong philosophy. So we have different themes. For example, in May, we will have supply chain as our theme. So we always change theme. We have, in a way, 2 or 3 conferences, all digital now. We plan to do face-to-face beginning 23. We hope that sincerely that this COVID will be out. But this Pharma Circuit, our vision is to really make it like the Google of pharma. Anything you come here, you will be able to see and you will be able to, do that. The trademark is, of course, registered now, and we are trying to, really scale it up and spread, across the world. In terms of business activities of this company, we do sourcing, nutraceuticals, food supplements. So I will not go through it. You can, look at it whenever you have time. And of course, we have found out a way, unique way, where, sourcing of pharmaceutical, nutraceutical, and food supplement creates an early access over the RX products, which takes 7 to 10 years of time. And why we are different? Because, everybody is doing networking, but we are trying to give a fair knowledge, which is theme based, and also make it special because of the, let's say, the market access and commercial and intellectual. So everybody is coming because it's absolutely free of charge. And that is the unique, nature of this conference. Of course, we find it, extremely, efficient to connect new ideas, and we can see that there is cross pollination happening, and maybe in future there will be something like Amazons of the world will, buy pharma circuit and try to get a platform for pharma, because Amazon, for example, is not yet in pharma. So I think I have just few minutes left for the talk. And before I end, I want to give you a positive message with 2 or 3, wordings. So first one, I like it the most, so the future can be predicted best when you create it yourself. I am too small of an individual. We all are contributing here, but I think we all worry about the future, and I also worry. But if we all stop worrying, it can be predicted best when we create it yourself. That means, you are the future. If you really think that you can make it, and you have the inhibition that, a solid instinct that you will do it, I'm sure you will be able to do it and make it big in the pharma world, because this is one area, pharmaceutical, which has never, let's say, seen any erosion, because there is always a need, and what best than, working with, academics. And another thing, which is my, another thought process, let's say it's not my word, but you should always try to have a different perspective. And that is the reason, if you see the slide, when everything appears to be going in one direction, take a long, hard look in the opposite direction. So I'm sure, these are not my words, these are all words from eminent individuals. But I found it quite relevant for my life, and for whatever I have done in last 20 years. And yeah, wish you all the best. Thanks for the opportunity. It's great to be here with you all. And with that, please contact me anytime you need any help, support. I am there for all of you. I'm grateful for being, there in Aurangabad and getting my degree from there. And I always remain close at heart with Aurangabad. Thank you for this opportunity.
Really very thank you, Dr. Kuntal Chatterjee, sir. Now the topic is open for discussion. Any questions from students, audience, please? Online ahead? Okay. No more questions, sirs. Really very nice, impressive and informative talk by Dr. Kuntal Chatterjee, sir. And it's very compendiously, Dr. Kuntal has covered from lab to market research. Also, how to approval a new drug, business development, and how to collaborate. Also, he's very nicely covered from small key points in terms of market strategies, and students will definitely be benefited after listening your talk, sir, in terms of what are the soft skills they have to develop during their MSc, and how to develop a long-term relationships in business, so that they will be benefited in their future, sir. So thank you very much, sir. Thank you very much. And we would like to listen you in our future event also, sir. Thank you very much.
Thank you to you all. Thanks to Hemaji for such an opportunity, and keep well. Thanks a lot. So bye for now.
Thank you, sir. Thank you.
Thank you.
Now, our next eminent personality is Dr. Joseph Selvin. Is he online? Soon he will join us in 5 minutes. So please wait for 5 minutes.
Are you there, sir? Joseph Selvin, sir? Yes. Are you there, sir? Can you
Yes,
hear?
I'm here, ma'am.
Yes, yes, sir. So next eminent personality is Dr. Joseph Selvin, sir. I would like to welcome Dr. Joseph Selvin, sir, from Department of Microbiology, School of Life Sciences, Pondicherry University, Puducherry. And welcome, sir. Welcome.
Welcome. Thank you.
I would like to request Dr. Hemalata Bhosle, ma'am, to please introduce Dr. Joseph Selvin, sir. Ma'am, please.
Welcome, sir. So it is again my proud privilege, and I feel very glad to introduce next eminent speaker, Professor Joseph Selvin, sir, Professor and Micron Support Project funded by European Union, and total outlay of his research is around 7.3 crores. He's been Fellow of National Academy of Agricultural Sciences, Academic Editor of PLOS One Journal, Jawaharlal Nehru Award recipient by Indian Council of Agricultural Research, DST Young Scientist Award, INSA Young Scientist Medal Award, INSA Professor LSS Kumar Memorial Award, UNESCO IOC Tema Grant Award, NPG Award 2006, DBT Young Investigator Project Award, INSA Anil Kumar Bose Memorial Medal, Member of Task Force in Department of Biotechnology, DBT MOES, Gujarat State Council and Kerala Biotech Council. He has been external reviewer of projects submitted under Cluster of Excellence Future Ocean in Kiel University. He is also a review panel member of American Association of University Women, that is AAUW. He has granted one US patent. One US patent is filed where technology developed based patent is there. Near about 15 research students have completed their PhD degree under her guidance, under his guidance, and around 3 postdoctoral students is mentored by sir. Around 150 research publications in journals of national and international repute with cumulative impact factor of around 435. 5 books have been published by sir, 16 book chapters to his credit, 19 proceeding chapters to his credit, and around 4,189 citations since 2017, with one recent research paper that has been published in Critical Reviews in Food Science and Nutrition with impact factor of 11.18. So such a great, eminent person is among us today. I feel very privileged, sir. I welcome once you again, and I request you to start your talk.
Thanks a lot, ma'am. Thanks a lot. So at the outset, I thank Hemalata madam, Dr. Hemalata Bhosle. So we have been associated maybe more than a decade. So this is our first opportunity we have interact each other, giving a talk. Probably I'm missing my presence there. Next time, definitely I will visit your university also. So I will get into the talk maybe another 30 minutes. I'll try to brief about the advances in synthetic biology. So anyone of the audience, I am audible, it is okay? Anyone of the audience, can you say, yes, I am audible?
Yes, sir, yes.
Audible, sir. Audible, sir.
Yeah, thanks a lot. Thank you. Okay. So actually, if you see the synthetic biology advances, it is going around all the field. The life science is actually entirely transforming. So I thought some of the insights I'll explain with the students and faculty here, so that you may take up some projects in the future. So particularly, I'll give you some insight about design and construction of endangered genomes, even aliens. So whether these are all we cannot imagine, but now it is possible. Okay, what is synthetic biology? By definition, synthetic biology is a multidisciplinary research. Definitely, it involving the principles of engineering. So we consider the organism or the human as a computer. So we feel we take the cell as the computer, not the whole organism. So the cell as the computer. Then we have hardware, cytoplasm as hardware, and the nucleus or the chromosome as the operating system. So that's the way they made the analogy between the cell and the computer. So if you see the development of synthetic biology, it is not a very old subject, probably a new emergence. However, this field is evolving from bioinformatics. So basically, it is starting from bioinformatics. Bioinformatics is evolving in the genomics. Genomics further evolved into proteomics. Then we know the analysis of metabolomics, and applying the mathematical models like our mathematical knowledge. Then we have systems biology, the system biology or systems biology evolving into synthetic biology. So now it may be 2010 onwards, people are working on synthetic biology, but we have a lot of applications and outcomes since 2020. Okay. So if you see the central dogma of life, everybody knows that. I'm sure you all have some, definitely you all have molecular biology knowledge, because biotechnology is the major theme of this conference. So gene, mRNA, protein, then metabolites and simulation. This is the way we are working. If you take the pool of genes, how we'll understand that is known as genomics. If you wanted to understand the pool of mRNA or transcript entirely of an organism, then it is known as transcriptomics. If you understand all the protein of an organism, including all the enzymes, hormones, etc., then the field is known as proteomics. If you wanted to understand the entire metabolic pathway of an organism, then it is metabolomics. But now we are using this terminology fluxomics. What is fluxomics? I will explain maybe after a few slides. So if you see the application of synthetic biology, you can see at the molecular level, even pathway or network regulatory levels, even it is at the organism level, even at the multicellular level. So organism means I'm indicating like unicellular organisms. So if you wanted to do some work, or if you wanted to start some work with synthetic biology, definitely I would like to introduce two software, plenty of softwares, because I deleted some of the slides. due to time constraint. So this is actually a web-based software. You need not install anything in your system, or you not required. You have a Java or other Linux or other platform knowledge, not required. So you can straight away start your synthetic biology work with GenoCAD. CAD means computer-aided design software. So where you can start, you can design your own genome, your own pathway, your pathway engineering. Everything is possible. So the second software I wanted to introduce is Tinkercell. So you can introduce or import a metabolic pathway, then you can delete some of the pathways to engineer or redirect the pathway to some other synthesis, synthesis of another molecule. What is metabolic engineering? How it could be performed? I'll explain maybe another few slides. Okay. So, see, we should understand the development of the synthetic biology is based on genomics. So, genomics is the basic. So, for the students' purpose, I will give you some basic information also. If you see, the cell biology is evolving into genetics, and genetics further evolving into genetic engineering. And genetic engineering further developed into cloning. You know that cloning of Dolly in 1996 was the biggest big news in all the media also. Then cloning, we have 2000 to 2003, another era known as genomics. Now also genomics era, no doubt, but that time we have the Human Genome Project dropped to us available. So the genomics evolving into genome engineering. Definitely, there is a strong difference between genetic engineering and genome engineering. Genome engineering further evolving into synthetic biology. So this way you can keep in your mind. So now the genomics, what you should understand, the facts of human genome are generally the term genome. The entire genetic makeup of an organism, that is genome. So very briefly, how you can keep in your mind, genome contains only 4 alphabets. AGCT, adenine, thymine, cytosine, and guanine. So the AGCT is repeatedly written, haphazardly arranged. That's all. That is genome. So a human is having 3.2 billion base pair, means 3.2 billion nucleotide sequences. So the arrangement is the matter. Okay, 3.2 billion sequence information, how it is arranged is the matter. Means, see, I would say, the genes are arranged in particular location, the gene loci. So that changes will have difference in expression. Yes, I would ask a question, because it's a lengthy lecture. I'm trying to run a little fast. So we all maybe 100 or 250 people are sitting here. So we are actually made up of same genome. There is no difference. Almost 25,000 genes, 19,500 real genes. Then why we are different in look and behavior, nature, etc. Why we have a diversity within humans? Because the gene loci. The gene loci decides that maybe you see even the color complexity. Someone looks little more pigmentation, someone is less pigmentation. So the melanin synthesis gene, where it is located, it is the matter. So means the location of the gene definitely makes the diversity. But otherwise, genomically, we are similar. Definitely no change. So if you find some of the interesting fact from the earlier research, in synthetic biology, particularly the one minimal genome. So I told you, a human is made up of 3.2 billion nucleotide sequence base pair. So if the 3.2 billion base pair is required for our life, definitely no. 97% of the genome do not have any function. We don't identify the function or what is its role. So that large proportion is nowadays we use the term junk DNA. Then can we create a human with 3% of our genome, which is have the functions and fingerprint area? So being this hypothesis, there was a research in 2005. So the publication indicates that we can delete the unwanted, and I should not use the word unwanted, okay. We can delete the uncharacterized or the functions unknown to us. We can delete that region. We can design a genome with only the known genes and fingerprint regions. Okay, that's a way. They've taken a bacteria known as Mycoplasma genitalium. It is a potential human pathogen, but it's a cell wall-less bacteria. So they've taken this bacteria, they deleted 100 genomic regions. So they identified 382 genes of totally identified, totally 482 genes they identified, but how much required is only 382 is sufficient for survival of Mycoplasma genitalium. Then the question is, if we delete the 100 genes which do not have any functional role, what will happen? They're using this global transposon mutagenesis is a technique. They deleted the 100 genes. So they created a minimal genome. Okay, minimal genome. What is that minimal genome? So this is this work was done by Hamilton Smith, a Nobel laureate. So this was the well demonstrated, I can say, the first synthetic biology work. So they deleted. Now what happens? You see, now they made a completely synthetic genome design. So the synthetic genome design of Mycoplasma genitalium shows that the first level you will find watermark. And second level and third level, they arrange transposones. Transposones, so it have a crucial role in evolution, as well as we can use as fingerprint region. Then synthetic DNA cassettes, then protein coding genes, then structural RNAs. Now carefully watch. If you know genomics or not, you should understand this architecture. In this architecture, see, this is a human-made genome, very important. It is not natural. The watermarks given in the first level, not available in any of the natural genomes. Then the transposon, 2006, 1999 means it is year of discovery. 2006, they identified some of the transposons. 1999, they identified some of the transposomes. See, this level of architecture arrangement also synthetic. It is not exist this way of arrangement in the naturally occurring Mycoplasma genitalium genome. And I told you 382 genes. Okay. Then if I ask you to arrange these 382 genes, what's the way you should arrange? You do not know. A to Z. Okay. But it is the genes are you have in a haphazard order. But how to order it in a genome, we do not know. Then somehow you know the reference assembly. Already existing genomic reference, you can arrange it. Definitely it is possible. While you are arranging the genes, now the question is, the 382 genes are arranged just like A to Z without any space between them. Definitely it is not possible. Definitely we provide space. Otherwise there will be no gene expression. So that space is known as synthetic DNA cassette. You understand me? Okay. So I'll explain one by one. The first level is watermark. The watermark is encoded with secret messages. Greg Winter designed this Mycoplasma genitalium synthetic genome. It is obviously known as Mycoplasma laboratorium. So this synthetic genome has a lot of secret messages they provided in the watermark. Like Craig Venter is encoded in this oligonucleotide sequence. Bender Institute is encoded in this oligonucleotide sequence. Hamilton Smith, the name is encoded in this sequence. Means once the synthetic bacteria, it is now available, which escaped into the nature, and over a period of time, another 50 years, another 5 decades, someone isolating the synthetic bacteria in the laboratory, while they are sequencing, they could identify that it is synthetic. The genomic architecture definitely reveals that it is a synthetic bacteria, not a natural bacteria. Then who has designed? Definitely this watermark will explain. Even after 5 decades, when hundreds of years, the discoverer may not be survived, but the people who are isolating this bacteria again from the environment or anywhere, they could identify who has designed this genome. I told you the DNA cassettes. DNA cassettes are spacer, like we are making lines, one line, then we are giving space. or each word we are providing space. Very similarly, they have provided space between the genes. That is 5 to 7 kb cassettes. Means that everything is oligonucleotide sequence only. Okay. If you see, in synthetic biology, we use the term biobricks. Biobricks means the part of the genome or part of the architecture. See, we can use a different part, maybe transposons, watermark, or DNA cassette, protein coding sequence, etc. But whatever may be, the naming is different, but everything is made up of oligonucleotide sequence only. Everything is oligonucleotide. So here, the parts means maybe the part of the genome. Device, for example, I would say one gene of metabolic pathway is a part. Then the entire metabolic pathway, we can use the terminology device. Then all the metabolic pathway is pooled. If your design is complete, then it is known as system. I told you, just we are considering the cell as the computer. Okay. So I will explain another 10 minutes, one example, how to design a cell factory. See, you might have known about fermentation or bioprocess technology. So here we consider the cell as the chemical factory. How? It is a single cell. We provide only carbon in terms of, I can say, paddy straw. So we provide only carbon as nutritional source. No other source, no other nutrition we will not provide. So the cell able to convert the carbon into all the required enzymes, metabolic intermediates, everything, as well as the cell to grow from this carbon metabolism. So using this metabolic pathway, the cell able to synthesize high value chemicals. So that's the way very economical fermentation and production methods we can be able to develop. Cell as the cell factory or chemical factory. See, what is the high value chemicals? Fuels, next generation fuels definitely depends upon a microbial cell factory. Engineered products, even plastics, polyhydroxyalkanoates or polyhydroxybutyrates, bioplastics. Why bioplastic is now not in the commercial scale? Because it is uneconomical. Be able to synthesize large volume of bioplastics in a cell factory, definitely it become normal use in everywhere. Commercial scale, definitely it is possible. Then bulk chemicals, vitamins, therapeutic pigments, amino acids. Then people are saying butanol. Butanol is considered as jet fuel. So all type of fuels, high value chemicals can be possible. But you should understand one fact. By genetic engineering, we cannot produce organic molecule. Some of the students have misconception that by genetic engineering, we produce any organic molecule. It is not possible. But by genome engineering, we are able to produce any of the organic molecules, which is not possible by genetic engineering. Okay, this is the real difference. So here, if you see, this is a traditional practice to increase the production of a particular, maybe an enzyme or a particular protein. What we will do, optimization. We optimize the environmental, nutritional, other factors. Then we perform mutation. But all these train improvement methods may increase the production tenfold. That is not sufficient for commercial scale production. Therefore, our idea is we need to redirect the entire metabolic pathway to the synthesis of a particular product. So the bacteria should not synthesize even any of the unwanted product. Only, for example, it is ethanol. Ethanol means ethanol. No other enzyme. Nothing should be released into the media. So that's the way the metabolic pathway should be redirected. So that process is known as metabolic engineering. So you'll find one example here. So this is a metabolic path. This is a metabolic pathway. An example. This is carbon source. A is carbon source. So in natural metabolism of a cell, a bacterial cell, what is happening? A is converted into product D through 3 intermediates. A is converted to B, C, D. So these intermediates are actually synthesized through the genes 1, 2, 3. Okay, this is a natural pathway. Now I wanted the product DE. Product E, the bacteria cannot synthesize. Then what I can do, I need to engineer the pathway, simply delete the gene 3. So there will be no product D. Instead, I insert the new gene 3 that secrete another enzyme, not the previous enzyme, naturally occurring enzyme, new enzyme 3, that releases a product D2, not product D. Now the product D2 is a metabolic intermediate, not a final product. So I need another enzyme which should act on this metabolic intermediate D2, thereby I will get the product, my expected product E, so that I'm introducing the gene 4. So gene 4 able to convert the metabolic intermediate D2 into my product E. You get me this point? This is known as metabolic engineering. But it looks simple, but it's not so easy, straightforward. So you can see one example, mucinic acid production in Corynebacterium glutamicum. Here, the red color indicates the additional pathway. So the blue color indicates the pathway deleted. So by the same time, we have to introduce new metabolic pathway and we should delete other pathway also. Otherwise, what will happen? Be able to redirect the metabolic pathway into mucinic acid, that is possible, but that may not be sufficient production you cannot achieve. If you want sufficient production, we need to redirect the pathway, by the same time delete the unwanted pathway. But by the same time, some issues are there. See, one simple example, methanol is converted into polyhydroxyalkanate, a bioplastic. So the methanol conversion undergoing two metabolic pathways, okay? So if either it converted into polyhydroxyalkanates or carbon dioxide. So I can decide, I can delete the second pathway, leads to carbon dioxide. Thereby, I can increase the production of polyhydroxyalkanate. That is possible. Theoretically, it is achievable. So if you delete this pathway, what will happen? See, you can delete the pathway. The formaldehyde, see, this is formalin or formaldehyde, is highly toxic to the cell. It is a metabolic intermediate. So the accumulation of metabolic intermediate will be toxic to the cell will die. So you performed very, very perfectly designed, perfectly metabolic engineering was achieved, but the metabolic intermediate accumulation makes the cell death, then our entire exercise will go waste. So this phenomenon, you have to consider the metabolic intermediate should be available in a particular concentration. That phenomenon is known as fluxomics. I told you what is fluxomics. Okay, so I'm leaving these are all. One more example. You know the artemisinin. Artemisinin is a plant-derived secondary metabolite, which cannot be synthesized in a bacterial cell. By using the same approach, what I explained, the metabolic engineering approach, which was successfully expressed in a bacterial cell. Now, artemisinin can be synthesized in a bacterial cell. It is an antimalarial drug, which was awarded Nobel Prize. You know that in 2015, the Nobel Prize awarded for the discovery of artemisinin. So this is artemisinin. It is actually originally this molecule was identified from a Chinese plant, Artemisinia annua. So if you see the metabolic pathway in the plant through the photosynthesis, which is converted to an acetyl-CoA, this is our playground. Yes, TCA cycle is the central pathway. Bacteria do not have this photosynthesis, chloroplast, nothing. But the bacteria is having acetyl-CoA. That's all in TCA cycle. That is our engineering route. Okay. So then it is converted to FEP, farnesyl pyrophosphate, amorphodiene, artemisinic acid. This is the pathway. But if you see, the bacteria is having only acetyl-CoA. All these entire pathway is missing. So we have to design the genes and the pathway, what is missing genes. Then if we are able to introduce the entire pathway into a bacterial cell, entire pathway means the genes what is not present in the bacterial cell. So if we are able to synthesize the missing genes and transfer the missing genes into a bacterial cell, the bacteria are able to secrete artemisinic acid or artemisinin. Now you have to carefully understand, in genetic engineering, we at a time, we clone only one gene. But in genome engineering, the subset of the pathway, a part of the pathway, completely designed, synthesized, and transferred to a bacterial cell. This is the unique difference between genetic engineering and genome engineering. See, you can see. So now we have taken off FEP, dimethylallyl pyrophosphate, inosine pyrophosphate. This entire pathway is transferred to a vector and transferred to a cell. Now the bacteria is synthesizing artemisinin. Now what you understand is synthetic biology is an excellent platform to synthesize plant-derived molecule. You know that the plant, we cannot depend on the plant to secrete that molecule because it's environmental stimuli dependent. So be able to synthesize all these organic molecules in the laboratory. Okay, already 20 minutes passed. I will go ahead quickly. So another application advances. So the synthetic biology advances is we can bring back the endangered animals. You know the mammoth. Mammoth is an like I said, the movies you might have seen. The elephant, the elephant family, which is not exist today, endangered, which is not exist. But the mammoth genome reconstruction project, clearly they achieved very recently, after 10 years of time, they completely designed and synthesized the genome. That is achievement. So they have only the tusk. The tusk was collected from the Siberia. Using this tusk sample, so maybe another 2 minutes, I able to, I don't know, 1 minute, 2 minutes, I'll run this video, then I'll quickly. So,
the scientific study has been recovered from mammoth remains up to 1.2 billion years old. It's 1,000 times older than Viking remains, and it even predates the existence of modern humans and Neanderthals. The analysis is led by researchers at the Center for Paleogenetics in Stockholm, revealed a previously unknown type of Siberian mammoth. We have recovered DNA from mammoth teeth that have been buried in the Siberian permafrost. This has been quite a challenge. It has taken us 7 years to get the data out and to analyze it. What we have found is that one of the specimens, which we refer to as the Krestovka mammoth, belonged to a previously unknown lineage of mammoth that we didn't know existed. And it also turns out that the Colombian mammoth, one of the most iconic Ice Age species of North America, is a hybrid between this newly discovered Krestovka mammoth and the woolly mammoth. Study published in Nature also managed to trace genetic changes across the species.
Okay. So this genomic information is now available in a journal. So in February 2021, they published this whole genome of a endangered animal. Now this project leads to bring back the mammoth to the earth because its own family elephant exists today. So if definitely another decade of time, I'm expecting it is successful, that would be a greatest breakthrough, be able to bring back the endangered animals not exist on the earth. Then the next project, I'll quickly summarize another 5 minutes. How to cheat death. So we have actually a definite birth date. Can we fix our death date? That is not possible. But now the projects are leading to be able to fix our death date. Definitely, I don't know whether it is true or not. One good example is, it is a 300 million project sanctioned to Great Winter. So what is the basic phenomena of hypothesis of this project? I will explain another 2 slides. See, now our lifespan is almost maybe 90s or 70s or 80s, whatever maybe. We do not have 3-digit lifespan. 3-digit means 100 plus. So if you see, 40% of the males who reach the age of 50, they won't make their age 74. Very similarly, 28% of the females, those who are now in 50, they won't make their lifespan to 74. Means, by nature, the female population have longevity when compared to the males. Okay, we actually, I'm belong to the male population. 50% of our population cannot make our lifespan up to 74. But in the case of female population, 28% of them cannot make their lifespan up to 74. This is quite nature. It is occurring now. So what Greg Winter predicts, one of his study, a 18-year-old girl, a patient, he identifying pre-diabetic, pre-cancerous genes in the genome. If you're able to identify, India is in the world, India is the largest burden of diabetics. Nowadays, the diabetics and hypertension, we cannot omit any of the houses. All the houses having diabetics and hypertension. So, if you identify the pre-diabetic genes and pre-cancerous genes at the early childhood, so Greg Winter is evolving some technique to silence or to delete that genes over a period of maybe after while we are growing, these genes get expressed, and these genes are the predisposing factor for the diabetics and cancer, are the two major life-threatening diseases. So this is the way. What they are predicting, all the diseases can be scanned at the childhood, even by birth, in our genome. If you're able to delete the genes, or if you're able to develop some methods to mitigate this gene expression over a period of time, we will escape from, maybe we can have longevity in our lifespan. This is an interesting project. In 6th September 2021, the Amazon founder, Jeff Bezos, funded another $270 million to this project. Again, another longevity project. Greg Winter already have a project. Now he invested heavily to have human longevity project. This include a Nobel laureate, a big team also. Even Sayana Yamanaka, he is a Nobel laureate. He is an expert in cell reprogramming. So their aim is, if they are successful, maybe Amazon will play a strong role in human longevity. They wanted to extend our lifespan at the 50 years of time. So these are all, the synthetic biology plays a great role actually, these projects. So that's the last slide, before the last part. See, Stephen Hawking, you know that. So we heard about Darwin's theory. So after Darwin, our time, the theoretical scientist we know is Stephen Hawking. So Stephen Hawking explains a lot of theories about aliens, the end of the earth, etc. So Stephen Hawking explains that a period of time, the space mission able to communicate with the aliens will conquer the earth, that would be the end of the universe. I'm not favoring that theory. So my question is, who is alien? We might have seen some movies, etc. We have some idea this may be an alien. But through this synthetic biology, I will explain you who is alien. The alien is, all the life on the earth is made up of AGCT, the 4 alphabet. Adenine, thymine, cytosine, and guanine. You cannot find any life other than AGCT. So you have a question, the tree of life, the phylogenetic tree speaks, all the life is related to each other. That's the reason the human insulin is, we are engineered to secrete in a bacterial cell. Bacteria is not diabetic, but we are secreting human insulin in the bacterial cell. So now the question is, other than AGCT, life other than AGCT is possible. Now, the expanded genetic alphabet. Genetic alphabet is 4, AGCT, that's all. Means semi-synthetic and synthetic genetic alphabet discovery is already successful. Once we create, already I start with my talk with synthetic bacteria. So the corona also, the COVID-19, the question was there, whether it is an engineered virus or natural virus. Still it is not resolved. Once we have a synthetic bacteria, synthetic virus with unnatural nucleotide, that is not natural. So once we created life with unnatural nucleotide, that is known as alien. So that would be the probably Stephen Hawking predicts the aliens will devastate the universe. Probably as a life science scientist, we can say that once we have the synthetic nucleotide-based life on the earth, that may be the devastating the natural life, including humans. So my concept is not my concept based on the literature and hypothesis is unnatural nucleotides and synthetic genomes that would be known as synthetic life. So with this, I conclude. Thanks a lot for listening. So we all see you all maybe another time we have to meet each other. Wish you all good luck. Thank you.
Thank you very much, sir. Thank you. Now the topic is open for discussion. Any questions from audience, please? Online.
Hello. Dr. Joseph. Am I audible?
Yes, sir. Yes, sir. Yes, sir. Please go ahead.
Yeah, it was nice talk. Yeah, we would have loved to listen your experiences and your research work from your lab in the synthetic biology you were talking about. But anyway, it was a nice lecture. And looking at the future of synthetic biology in India, we are not allowing transgenics to be released, even though it was developed. And then we are talking about synthetic biology. So what do you think? What is the future of this synthetic biology in India? Another thing you mentioned, Craig Venter, I was fortunate to meet him when he was in India. And then we saw a lot of work, and he was talking about this mycoplasma genitalium, and he converted it to laboratorium by genome swapping, and now he is working on all these technologies. So thanks, we have people like Venter. But could you please just tell us what could be the future of the synthetic biology in India? So what is your opinion?
Synthetic biology actually, DBT is giving actually IBEC. IBEC means a competition among the students. Already DBT started funding. So if the students give some project idea, they are funding 10 lakhs initially, then they're giving 50 lakh project. So India already they started funding. Definitely I expect now the work is going on genome engineering. So definitely India cannot escape. India is supposed to grow on synthetic biology, but unfortunately we are in the very negligible output from the Indian side. But I'm sure we will also compete with other countries with synthetic biology work, but it requires a lot of funding. Not so easy. Like we cannot do any basic work without funding. But one interesting future is we can do a lot of work with in silico side. That have a lot of opportunity, not require any funding with the students, even small colleges can start work with synthetic biology, but many are not started working.
Thank you, sir. This is another question.
Yes, sir. Yes, sir.
Thank you for nice presentation. What if this synthetic bacteria, genetically modified microbes escapes into the nature? What the suspected case happened with COVID-19?
Yes, sir. This is actually this will be maybe the future. Definitely based on the bioweapons, the warfare. Definitely based on synthetic bacteria and synthetic viruses. I'm sure maybe another decade of time people discuss about the transgenic escape, the escape of the synthetic bacteria in the environment. Definitely, it creates a lot of troubles. Though all the projects are leading to, because the vendor designed the mycoplasma laboratorium for the enhanced production of fuels. So all the projects aiming a good output, definitely. That leads to definitely a challenge to the diversity, biodiversity, even humans, and natural phenomena of existence. So I don't know, because you all be with us, maybe we may wait for another decade of time. What is the impact of synthetic biology on the earth?
Thank you very much, sir. Thank you for your informative and impressive talk on synthetic biology approaches for synthesis of natural products in microbial cell factories. Sir have covered very nicely biobricks, then strategies to improve strain for commercial production, Then fluxomics, very systematically, mannerly, sir has presented a topic. So thank you very much, sir. And thank you very much for accepting our invitation in spite of your busy schedule, sir. We are expecting you physically in our future events, sir. We would like to listen you again in future, sir. So thank you very much, sir. Thank you
Thanks a
very
lot.
much.
See you. Bye bye.
Thank you, sir. Now we'll have lunch break, and for 2:15 p.m. we will be here for our next session. So lunch is arranged at School of Life Sciences. So enjoy the lunch. Sharp timing at 2:15 p.m. Savadon.